So good morning, everyone. Thank you so much for joining us for the fifth annual TIC Academy. Since 2013, the TIC IPM Working Group has worked to organize and expand the network of diverse stakeholders working to reduce the risk of exposure to infected ticks by collaborating on IPM-related activities, exchanging knowledge, and sharing resources. The TIC IPM Working Group has done a lot of work to put on the TIC Academy, and we are very, very excited to bring you this year's edition. The TIC IPM Working Group is run by the IPM Institute of North America, where it has been working for over 25 years to increase adoption of the integrated pest management and improve sustainability in agriculture and communities through a variety of programs. Our organization has two sections: Sustainable Food Group and Sustainable Communities Group. The TIC IPM Working Group is part of the Sustainable Communities Group. Sustainable Communities Group has three major projects: Green Shield Certified, which we have for pest management providers, facilities, and landscapes; the Pest Defense for Healthy Schools; and of course, the TIC IPM Working Group. If you would like more information about any of these programs, please feel free to reach out to either myself or Josie Dillon, who is here today helping with technical support and helping to moderate our chat and Q&A for most of the day. So now would be a good time to put some perfect contact information in the chat there for you. The TIC IPM Working Group has a website that we would love for you to visit. It is in the process of being revamped to be a little more user-friendly and a little more accessible compared to what it has been in the past. It's important to note that this is a section of the IPM website and not the TIC IPM Working Group page that shows up on Google. That's an old website that we're in the process of getting rid of. But please visit our website to learn more about the group. We host webinars on the second Wednesday of every month at 1 p.m. Central Time. They're about an hour long and cover a wide range of TIC-related topics. We bring in all kinds of exciting speakers, some of the presenters that are here today and tomorrow have presented at webinars before, and people have liked them so much that we asked them to tell us more at the TIC Academy. Very excited about our monthly webinars, and we hope that you will join them in the future. If you are interested in joining the working group to receive information about webinars, please reach out to me. My email's there on the screen. We can put it in the chat as well. Or if you or someone you know is interested in presenting at a future webinar, please let me know. We're always looking for new presenters for future webinars. And related to our webinars is our YouTube channel, which is also in development and will continue to get better. But this has a bunch of our previous webinars. It has all of our webinars that have happened in 2024 and in this funding cycle. We have old webinars that I will upload to this, and all future webinars that will have recordings will end up here. So you can go to our working group YouTube channel by visiting our website and navigating to the channel that way, or by searching at TIC IPM Working Group on YouTube. Getting more into how today is going to work. We have continuing education credits for pesticide applicators from Michigan, Oregon, Texas, and Washington, and we have vector CEUs available for California. If you are here attending for CEUs, please email your state and license number to rnolan@ipminstitute.org, and I will make sure that we have the necessary information to get you credit for those presentations. This is our agenda for today and tomorrow. We have such an exciting lineup of speakers that we're really excited to get to in just a second here. But I do want to pitch real quick that if you're here today and you don't know that there's more tomorrow, please take a second to write down that you should register for tomorrow, or to do it right now while we're thinking about it. I'll tell you more about tomorrow later. But here is the lineup for today. There will be a poll question at the beginning and end of each presentation. Please answer to the best of your ability. Especially the question before each presentation. We're not expecting you to know the answer necessarily. If you do, that's great, but don't stress about it too much. Just select whatever option makes the most sense to you. And for the most part, you should learn the answer to the question by the end of each presentation, to answer it at the end. Audience participation is highly encouraged. We want you to get the most out of this. So if you have questions for our speakers, please put them in the Q&A function, which is important to know, is separate from the chat. So in Zoom, there's the chat and there's the Q&A. Please use the chat for general questions for myself and Josie Dillon and other IPM staff, or general comments, and use the question and answer function for questions that are intended for the speakers. Josie Dillon from the IPM Institute is online today to help monitor the chat and answer questions. And Alexandra Maresco from Project Lime will be helping to moderate the Q&A for our keynote presentation, which is coming up in just a second here. There will be a 30-minute break around 11:30 a.m. Central Time. And lastly, on structure, this is being recorded both today and tomorrow. So if you are here today and you want to watch this again, you will be able to do so. If you are only here for part of the day and want to catch up on the afternoon, or are here today and have registered tomorrow but are going to miss it, there will be a recording and you can review all of this at a later date. If you have any questions, please feel free to email us. And finally, we want to thank our sponsors for making this possible. We have 3 levels of sponsors, and really, it takes so much work to put this on, and the sponsorship is very helpful, and we want to take a minute to recognize them. So our gold sponsors are Tickbox Technology Corporation, who you'll hear more from later, Project Lime, US Biologic, and mainly Ticks. All of our sponsors you'll hear more from later. We have some information about them later on. Our silver sponsor is the North Shore Mosquito Abatement District, and our bronze sponsors are Alexis Chesney, MS, NDLAC, and Central Life Sciences. So thank you so much for your support. We're really excited to show you the work that we've put into this. And the primary funding for the TIC IPM working group comes from the North Central IPM Center, which is the TIC IPM working group is supported by the USDA National Institute of Food and Agriculture, Crop Protection and Pest Management Program through the North Central IPM Center. We are currently in the process of applying for future grant support from NCIPM, and if you would like to support the work we do, please consider writing us a letter of support. For more information to receive our letter of support template, please contact me or Josie Dillon, and we will get you more information on our current needs for this grant cycle. So with that, I'm going to switch over to our first presentation. Bear with me just a second here. Can we see the presentation?
Looks good.
Wonderful. So I'm going to, Josie, can you launch the poll question? And while she does that, I am thrilled to introduce our keynote presenter, Dr. Laura Kahn. For almost 20 years, Dr. Laura H. Kahn was a research scholar with the Program of Science and Global Security at the Princeton School of Public Health, Public and International Affairs, Princeton University. Her education and training encompasses nursing, medicine, public health, and public policy. In April 2006, she published Confronting Zoonosis, Linking Human and Veterinary Medicine in the Centers for Disease Control and Prevention, CDC, Journal of Emerging Infectious Diseases, which helped launch the One Health Initiative, a global movement promoting the health of all species by increasing communication and collaboration between human, animal, plant, environmental, and ecosystem health professionals. Dr. Kahn is the author of several books. The first, Who's in Charge? Leadership During Epidemics, Bioterror Attacks, and Other Public Health Crises, was originally published in 2009 by Prager Security International. In 2020, a second edition was issued with a new preface discussing leadership during the COVID-19 pandemic. Her second book, One Health and the Politics of Antimicrobial Resistance, was published in June 2016 by Johns Hopkins University Press. Her third book, One Health and the Politics of COVID-19, will be published in October 2024 by Johns Hopkins University Press. A native of California, Dr. Kahn holds a bachelor's degree in nursing from UC Los Angeles, a doctorate in medicine from the Mount Sinai School of Medicine, a master's degree in public health from Columbia University, and a master's degree in public policy from Princeton University. In 2014, the American Association of Public Health Physicians awarded her with the Presidential Award for Meritorious Service, and in 2016, the American Veterinary One Health Society awarded her with the highest honor, the K.F. Meyer James H. Steele Gold Head Cane Award for her work in One Health. Dr. Laura Kahn.
Thank you so much, Ryan. It's really a pleasure for me to be with you this morning and to talk about give you a One Health perspective on Lyme and other tick-borne diseases, as well as ticks. Now, you all are the tick experts, so I don't claim to be an expert on ticks, but I am well-versed on One Health, which I will share with you today. Next slide, please. So you may or may not have heard about One Health, the One Health concept. Very simply, it's human, animal, plant, environmental, and ecosystem health are linked. And this concept is useful as a framework to examine complex issues such as the zoonotic diseases, vector-borne diseases as well. And if we're to develop effective policies to address these diseases, we need to look at their root causes. And I think it's important that we recognize that we interact with our environments and our ecosystems every day, not only by going outside or living in our indoor environments, but we're literally incorporating the environment by inhaling air, drinking fluids, and eating the plants and animals that we call food. And you see at the bottom of the slide, the One Health Initiative website. It's a labor of love for my colleagues and myself, so please visit it and tell your colleagues to visit it as well. Next slide, please. So One Health has been visualized in a number of different ways. In the upper left-hand corner, you see it's depicted as being coordination, communication, collaboration between different disciplines. On the upper right-hand corner, you divide it out by animal type, whether it's wildlife or domestic livestock, with human health, One Health in the middle. On the left-hand side, humans, left-hand bottom, animals, humans, environment. And then in the right, lower right, you've got the umbrella graphic with zoonotic infections in one bubble, chronic diseases in another bubble. One Health started as an effort to control zoonotic diseases, but it has evolved over time to include many other subjects. So it's very broad, but it's versatile and very applicable. Next slide, please. So when I think about One Health and how I use it for my research, I think of it as a multidimensional matrix, or a Rubik's Cube, if you will. And along one dimension are the humans, animals, plants, environments, and ecosystems. Along another dimension, it can be complexity and scale, microbial cellular level, individual, and then population level. And then you can include the political, social, and economic factors at the political border level, local, regional, national, and even international, global. Next slide, please. So if we smush down that cube and make it two-dimensional, then you can look at all the intersecting boxes, if you will. And you can then slice it and dice it depending on your needs, on how you want to examine an issue. Now, for this talk, when I talk about environments, I'm talking about the abiotic, the soil, the water, the air aspects of a defined geographic area. And when I talk about ecosystems, I'm talking about the biotic interactions, the microbes, the flora, and the fauna within a defined geographic area. Next slide, please. So in this talk, then, I'm going to take some of those intersecting boxes that I showed you in the previous slide, and I'm going to very systematically and comprehensively go through each one of these in turn, and hopefully provide you with a broad-based perspective on tick-borne diseases. So in a sense, I'm throwing out a wide net, and I'm pulling it in, and I'm seeing what I come up with. So that's kind of how I approach One Health. Next slide, please. So let's look at our first little mini analysis, looking at humans, animals, domestic, and wildlife. Next slide, please. So in researching this talk, I found this interesting slide, and I must admit, I did not know that ticks are part of the arachnid class. So they're along with spiders and scorpions. Who knew? You learn something new every day, and I certainly did when I found this slide. In this talk, we're going to focus on the Ixodidae, or the Ixodes. Maybe I'm not pronouncing it correctly. Correct me if I'm wrong. The ticks. There's over 900 tick species, so there's lots of ticks. But the ones that I'm going to focus on are the ones that are the primary, serve as the primary vector for Lyme disease. Next slide, please. So the Ixodes ticks, is that how you pronounce it? Sounds like Ixodes. Okay, thank you. They're known as the three-host ticks. You've got the rodent reservoir, which I'll talk a bit more in a few slides. The eggs of the ticks are not infected. It's only really when they start feeding on the rodents that they become infected. Humans are an inadvertent host. And then the adult ticks like to feed on large mammals like deer in that stage. And tick attachment can last anywhere from less than an hour to 7 days. Next slide, please. So tick-borne diseases are global. The focus of this talk is going to be on Lyme disease in the United States. It's a serious problem in the US, but it is important to recognize that these are global, and these diseases are global. And the pathogens affect both humans and animals. In terms of animals, both domestic and wildlife. Next slide, please. So let's look at, do a comparative Lyme disease epidemiology between the spirochetes, the humans, and the dogs in the United States. Next slide, please. So this was a study done looking at the distribution of Lyme disease spirochetes in the US. And you see along the Pacific coast and along the eastern seaboard and going into the middle of the country. But what was most striking to me about this map is the big kind of glaring gap between one side of the country and the other. And the question is that because these ticks just weren't being sampled, or there's just no infected ticks in the middle of the country. And that's an important question clinically, because if there are no infected ticks and somebody comes in with Lyme disease symptoms, then the doctor is going to say, well, there's no Lyme disease in this area, so there's no way you can have it. So keep that in your mind as I progress through this talk. Next slide, please. So when you look at human cases collected by the CDC, 2022, you can see Lyme disease is hyperendemic in the Northeast and in the upper Midwest. Over 63,000 human Lyme disease cases were reported, but the CDC estimates 476,000 cases were treated for Lyme disease. So it's a serious problem. Next slide, please. Now, this is canine data. Canine data, aggregate canine data from 2012 to 2016. From over that period of time, over 1 million lab tests for dogs were tested positive with a seroprevalence rate of about 6.4%. And what's interesting about this graphic is it kind of fills in some of that blank space that we saw with the infected ticks and the gap in humans. There seems to be a lot more Lyme disease around the country with dogs. Now, granted, there is still some blank areas in the middle of the country, but there's a lot more there than what we would think if we were just looking at humans. Next slide, please. So there was some models that were done using that data, and they kind of filled in the entire country. So yes, it's hyperendemic in the Northeast and upper Midwest, but there does appear to be some canine Lyme disease in other parts of the country. Next slide, please. This is compared to the human Lyme disease incidence, where again, that huge blank area in the middle of the country, suggesting that there isn't any. Next slide, please. If you compare the canine with the human, I think one could argue that you actually get better resolution of what's in the environment through the canine data than with the human data. Next slide, please. So dogs are tested annually for Lyme disease. It's just part of the regular health care provided for the animals. Next slide, please. So because of that, I think we get better resolution. And from 2011 to 2015, the IDEXX labs collected nearly 12 million Lyme disease test results from veterinarians, and they determined that there are 50 times more dog cases of Lyme in the South and West than human cases, 25 times more dog cases in the US overall. So the question is, perhaps we should be using canine incidence data for understanding Lyme disease in the US than humans. Next slide, please. So the canine cases greatly outnumber the human cases, as I had mentioned in the previous slide. Next slide, please. So dogs have a much greater proclivity for tick exposure. They're running through the woods and they can be covered with ticks. Ticks on a human, usually not so much. Next slide, please. And there's, from this one study in emerging infectious diseases, you see a very strong correlation between canine seroprevalence and canine seroprevalence for Lyme and annual Lyme disease incidence in humans. Very strong correlation. Next slide, please. Animals have been used as sentinels historically. So for example, the canary in the coal mine is a classic case. to monitor carbon monoxide poisoning. The dancing cats of Minamata Bay in Japan served as sentinels for mercury poisoning, and they helped to identify why people were getting sick and dying. Bald eagles in the natural environments can get lead poisoning if they consume an animal that has bullets in it. They get lead poisoning from lead bullets. Next slide, please. The animals have been useful sentinels for disease surveillance. So crows were dying by the thousands during the West Nile virus outbreak in New York City in 1999. Prairie dogs served as sentinels for the monkeypox outbreak in the US Midwest in 2003. And wild waterfowl serve as sentinels, and they help spread highly pathogenic H5N1 avian influenza in China in 1996. Next slide, please. So let's shift gears a little bit on the role of biodiversity in potentially protecting humans from Lyme disease exposure. Now, I had mentioned earlier in a previous slide that infected rodents are the hosts that usually infect the ticks. And indeed, it appears that white-footed mice are very competent reservoirs for infecting ticks. And if you have an environment or an ecosystem that's primarily mice, you're going to have more Lyme disease and other tick-borne diseases than in more biodiverse areas where you have hosts that are lesser competent reservoirs for ticks. In the right-hand side, you can see that with a computer model, the incidence of Lyme disease, the number of host species, as it increases, the percentage of infected ticks decreases. Next slide, please. Again, mice and other rodents, particularly mice, are extremely competent reservoirs for ticks, for the spirochete. And as you go down this list, they become less and less competent. So, for example, skinks are much less competent than mice. Next slide, please. So again, forest fragmentation, mice, and Lyme disease risk. Mice love fragmented forests, they thrive. And so when their population explodes, so does the Lyme disease risk. Next slide, please. So in other words, we should try to replace white-footed mice with skinks to reduce that Lyme disease risk. Next slide, please. Just quickly, this mini analysis looking at the plants and the trees. Some are repellents for ticks, some are attractants. Next slide, please. The power of plants. Some plants naturally repel ticks. Those like lavender, garlic, mint that have strong compounds or odors repel ticks. And some plants attract ticks, trees with low-hanging branches, deciduous trees. I'll talk a bit in a minute about that. Mulch, leaf, and grass litter attracts ticks. Next slide, please. So tick densities are highest in areas adjacent to deciduous structure-rich forests, and they're common in temperate areas. In contrast, mosquitoes are most common in tropical regions, although we certainly suffer from them in the temperate regions as well. Next slide, please. So again, this image shows forest interior, deciduous, structure-rich, dense with ticks. The coniferous, structure poor are not as conducive to high tick concentrations. Next slide, please. So the different tree categories, the deciduous, they have the broad leaves that change color in the fall that we enjoy so much. They lose their leaves. You get a lot of soil coverage by the leaves that the ticks like to hide in, as opposed to the evergreens. that are not as conducive to ticks. Next slide, please. And this is, I'm sure you are well aware, the temperate deciduous forest. That is like tick central, that the mice like and the ticks like. Next slide, please. Let's move on now to environments and ecosystems. Again, environment is the abiotic aspect of a geographic area: soil, air, water, climate. Ecosystems, the biotic aspects. Next slide, please. So climate change is a concern for many reasons, but one is that it will likely affect ticks and tick-borne diseases, expanding into new areas. Next slide, please. So to truly understand climate change, you need to think like a geologist. And if you're interested, I highly recommend this book, Timefulness by Marsha Bjorn. I can't pronounce her name. But anyway, if you're interested in this, I highly recommend her book. But the planet was very hot during the Paleozoic era. And there was thriving life in the seas, but the land was barren. And over millions of years, the planet began to cool. In the Pleistocene, it was the Ice Age. And inexplicably, right at the border between the Pleistocene and the Holocene, you can see that the temperature of the planet began to increase. We don't know why. But during the entire period of the Holocene, the era that we live in now, you can see that the climate has been remarkably consistent and stable, and has allowed agriculture and civilization to exist with the tiny deviation of the Little Ice Age, where it was about 1 degree below the Holocene baseline. We are now about 1 degree above the Holocene baseline, and we are starting to see tremendous impacts both in the storms and in its effect on food production. So I am sure that we're going to be seeing an impact on vector-borne diseases as the climate changes. Next slide, please. So in terms of environments and ecosystems and Lyme disease incidence, next slide, please. You can see the forest fragmentation of the deciduous forests on the upper northeast and in the Midwest, and that corresponds with the high hyperendemic Lyme disease. Now the question is, the middle of the country where we saw all those, the blank area, was it because of the evergreens, the types of forests and the trees that we have in the middle of the country? I haven't seen studies on that, so I can only speculate that perhaps that might be playing a role in why Lyme disease is not as common in the middle of the country, although again, the canine data suggests that there is some infected ticks regardless. Next slide, please. Moving now to the political, social, and economic analysis. Unfortunately, Lyme and other tick-borne diseases are highly political issues, and they are often associated with medical errors. Next slide, please. I don't know if you've heard the term the Lyme Wars, but there's been a long-standing controversy about Lyme disease and how to treat it and how to identify it. Next slide, please. So part of the problem with Lyme disease is, as we saw with some of those previous maps, the areas where there doesn't appear to be a lot of human cases, a lot of the physicians will conclude, well, we just don't see Lyme disease in this area, so there's no way that you can have Lyme disease. And they might even refuse to test the person for it. In many cases, people do not come in or do not recall seeing a rash. They don't recall a tick bite. I can attest I've had Lyme disease 3 times. I never had a rash and I never recall seeing a tick bite, but I had severe Lyme disease. So I personally experienced this situation where the docs just don't believe that you have Lyme disease. So it's become a very political issue, not only in the challenge of diagnosis, and the tests are problematic as well, but the treatment is problematic as well, because sometimes, even despite getting antibiotic treatment, you can have long-term sequelae and then getting late or chronic Lyme disease. Next slide, please. There's a number of possible mechanisms for the proposal of chronic Lyme disease, or why the treatment has failed, whether it's a persistent infection despite the antibiotics, or immune evasion, or maybe autoimmune dysregulation. We simply don't know. Next slide, please. So chronic Lyme, again, persistent infection. There can be damage to the cardiovascular system, the immune system, the nervous system. All of those things are possible with this pernicious disease. Next slide, please. And Lyme disease is extremely expensive, and by one estimate, the cost can exceed over 75 billion per year, primarily in people who are suffering from chronic Lyme disease and are seeking treatment for that. Next slide, please. So a brief recap in our One Health analysis, looking at humans and animals, plants, environments, and ecosystems, the political, social, and economic factors. Next slide, please. From a One Health analysis, Lyme disease causing spirochetes exist around the country, particularly in the Northeast and the upper Midwest. I would argue that the canine Lyme disease cases reflect a distribution around the country better than the human cases. There are more canine Lyme disease cases than human cases, particularly in the South and the West. Animal health has served as an important sentinel for environmental and human health. And I argue that canine Lyme disease surveillance data should be an important adjunct to human Lyme disease surveillance data going forward. Biodiversity, forest fragmentation, and the type of forests appear to be an important risk factor for Lyme disease. White-footed mice are extremely competent hosts for the spirochete, and they thrive in fragmented forests. Skinks and other lizards are very poor hosts for the spirochete, and they predominate in the South. Maybe that's why, one explanation for why there's less cases in the South. The political, social, and economic costs of Lyme disease are substantial. And a One Health approach, integrating human, animal, plant, environmental, and ecosystem health are important for us to try and get a handle on this situation. Next slide, please. So One Health recognizes the interconnections of life, and I hope this presentation has shown you that. And again, canine Lyme disease incidence data might be a very useful adjunct and perhaps should be part of our national strategy to address Lyme and other tick-borne diseases. Better land use policies, trying to avoid forest fragmentation, might reduce the environmental and ecological conditions promoting the spread of infected ticks. And our public policies must be interdisciplinary in using a One Health approach. Next slide, please. These are my colleagues in the One Health initiative. Next slide, please. And I'd like to thank you all for your time and attention. And next slide, please. And for those who are interested, the references that I used for this talk, next slide. Human tick-borne diseases and advances in anti-tick vaccine approaches, a comprehensive review. Next slide, please. Quantifying the relationship between human Lyme disease and Borrelia burgdorferi exposure in domestic dogs. That was published in Geospatial Health in 2019. Next slide, please. Canine serology as an adjunct to human Lyme disease surveillance and emerging infectious diseases. That was published back in 2011. Next slide, please. PLOS biology, why Lyme disease is common in the northern US, but rare in the South. The roles of host choice, host-seeking behavior, and tick density. Next slide. Missing the people for the trees, identifying coupled natural human system feedbacks driving the ecology of Lyme disease. That was published in the Journal of Applied Ecology. That was back in 2019. Next slide. The impacts of climate change on ticks and tick-borne disease risk, annual reviews. That was published in 2021. Next slide. And I think the power of plants and tick prevention. I think that's the last one. Next slide. And that's it. Yeah. So thank you all for your time and attention, and I'm happy to take any questions.
Laura, that was wonderful. And as an 8-year Lyme patient who went 2 years undiagnosed for exactly the reasons you listed, I'm very, very grateful for your work and contributions to this space. So for any of our participants, feel free to hit the Q&A button and ask any questions that you may have for Laura, and I will moderate. Well, I'm going to start with one that was left in the webinar chat.
Sure.
Someone asked why there isn't annual testing for Lyme disease in humans, and even in the endemic region where I live, she has been refused testing.
Well, that's part of the politics, unfortunately. Well, that's a good question. I mean, I think in the hyper-endemic region, people should probably get tested annually. Of course, there's going to be costs. What I didn't talk about is that, unfortunately, the diagnostic tests for Lyme disease are not very good. So in terms of screening, they'd probably be even worse. And part of the problem is it's an antibody test, so it's an indirect test. So it takes time for the body to build up antibodies to an infection. And so this test, if you test too early, then it's going to be a false negative because the body has not had a chance to build up the antibodies. For somebody who's had Lyme disease in the past, the test will not necessarily tell you if you have a new infection, because once you have antibodies, you're going to always have antibodies. So as a screening test, it's probably not going to be very good to tell you if you have an acute infection. But they do it for dogs. So so maybe our dogs are getting better health care than we are, and there's a vaccine for dogs. I have read cases where our canine companions are getting better health care than we are. And perhaps tick-borne, particularly Lyme disease, is one such example.
Yes, I completely agree with you.
Yeah.
We have another question in the Q&A asking if you have any thoughts on the West Coast Lyme disease symptoms where the Western fence lizard was shown to increase the density of infected ticks. Should we consider a host composition rather than just a good host versus bad host?
Yeah, if that's been shown to increase I would be very interested in that because, usually you would expect a reptile to not be as good a host, although, you guys are the tick experts, correct me if I'm wrong. But if it's been shown to be as effective a reservoir as the white-footed mice, then certainly, you're going to want to have to address your ecosystem in a way similar to how we have to address the white-footed mice. Now, what I didn't talk about was, well, how do we address the explosion of white-footed mice? If we want to try and control them naturally, then you want to have the natural predators that eat white-footed mice. So we'd want to promote owls and other raptors that depend on mice. I mean, part of the conundrum with Lyme and dealing with this is trying to control the disease without messing up the ecosystem. Because if you start using pesticides to kill off the mice, well, the owls and the other raptors and the other wildlife that rely on mice for their food, they'll get poisoned, and then they'll get sick. And then you start getting this vicious cycle of messed up, food chain, and dead and dying predators that rely on the mice. But we want to get rid of the mice because they're the reservoir host for infecting ticks. So it's a real conundrum on finding a good balance to make sure that you maintain a healthy ecosystem, because we want biodiversity. I don't know how much effort there is in trying to figure out how to control mice in a ecologically friendly way, relying on the natural predators.
Very interesting. Somebody also asked, Laura, if you have any thoughts on which antibiotics are effective.
Well, usually it's the doxycycline, the ceftriaxone. Those are usually the standard of care. I'm not aware of resistance being developed against them. So theoretically, they, a course of, and now that's the other conundrum, is how long do you need the course of antibiotics? And usually they recommend 2 weeks. But as we've seen, there are people who are failing treatment, and there are some physicians who are recommending prolonged treatment. Now, the other problem is with prolonged treatment. I mean, we rely on our gut microbiome and on a healthy microbiome for our health. And if you start taking months of antibiotics, you're messing up your microbiome. So when I talked about the ecosystem and how we don't want to mess it up by killing off all the mice and removing the food supply for the owls and the other predators that rely on them, well, along analogous lines, prolonged antibiotics is going to screw up our microbiome in ways that can have prolonged adverse health effects. So, these issues are not just, black and white. There's a lot of nuance here in trying to figure out how to address it in an ecologically friendly way in our own body, to get rid of the spirochete without completely messing up our microbiome. So it's a real balancing, and without trying to, create resistance. So it's a real conundrum, a real challenge. Disease.
Somebody else asked a great question. How would you compare vector-borne diseases that come from wildlife, like Lyme disease that requires a tick vector, to spill over diseases like bird or swine flu in a One Health perspective? Are they fundamentally different?
Well, with a vector-borne disease like ticks or mosquitoes, I think of mosquitoes like flying hypodermic needles. They're flying around and they're infecting you. The tick directly infects you. You get bitten, and it injects the pathogen directly into your body. So that's a type of spillover, one could argue. When we talk about spillover, and in my most recent book, which has just been published, One Health and the Politics of COVID-19, it's now available. When you look at spillover for something like SARS or MERS or COVID, the criteria there is very different than getting bit by a tick or mosquito. There, if it's a non-vector-borne disease, usually you have some sort of interaction between the infected animal and the animal worker. So, for example, with SARS-CoV-1 that came out in 2002, 2003, you had antibody and viral data from the animals. In this case, it was a civet cat, 99.8% identical to the antibodies in the humans. And it was the animal workers in the wet market in the lab in the Guangdong province. So you had the matching serologies, and we don't know if the virus was aerosolized or if maybe it was somebody cooking with the animal, cutting it, but somehow the virus gets either you inhale it or you ingest it, is the usual mode of a spillover of a non vector-borne disease. In the case of MERS, you had infected dromedary camels, and there was good serologic data of these camels going back decades, showing that they had antibodies to the MERS virus. And it wasn't until 2012 that it somehow spilled over into either the slaughterhouse workers or the camel workers. They had very high incidence rates of antibodies to MERS. So they proved through the matching serologies of the camels and the workers with the camels showed that spillover. You need that matching serologies to prove that. With COVID, however, there's no animal data. Nothing. There was a study done on the animals in the wet market in Wuhan. They tested 457 animals, many different species, 0 tested positive. At the same time, there's no occupational data on the humans. So we don't have the data proving a natural spillover with COVID. There is, however, circumstantial evidence that it might have come from a laboratory. So that's another whole talk. I have a talk for that, but anyway, that kind of shows you the difference. And the challenge with the vector-borne is not everybody remembers getting bit by a tick or by a mosquito. So, but it is essentially a spillover, but in a different mechanism. It's a direct infection rather than either aerosol or ingestion. But again, One Health, we're interacting with our environment every day. We're inhaling the air, we're drinking the fluids, we're eating the plants and animals that we call food. And if they're infected, we're going to get sick.
Mm-hmm. Absolutely. It makes sense. It makes so much sense that everything is interconnected. One of our last few questions that I'm seeing towards the end of this session is, for plant interactions, would the regrown deciduous forest be more fragmented compared to the Stone Age forests on the West Coast? There probably is a threshold for where certain host compositions can increase/decrease infection prevalence.
Yeah, you would think so. Unfortunately, the old growth forests like the redwoods, I mean, they've been cut down. There's like a tiny fraction of the old growth forests left on the West Coast. So they're pretty fragmented too, unfortunately. So you've got fragmented forests on the East Coast, and I would argue you've got fragmented forests on the West Coast too. Particularly the redwoods. Now the redwoods are evergreens. They're not deciduous. So theoretically, in those areas, one could argue that you might see less tick density than in a deciduous forest. But I don't know if people have looked at compare and contrast that. But you definitely have fragmented forests on the West Coast too.
Very interesting. Last question. Someone asked, how long after you're bit by a tick does that secretion stay on the human body? I guess it would really be in the human body.
Yeah, I mean, the tick, in a previous slide, the tick can stay attached from anywhere from less than an hour to, I don't know, 24 hours or more. So, you know, how long it takes for the tick to actually inject the spirochete into you, I'm not sure it's really been well understood, as far as I can tell. So, and again, most people don't know they've been bitten. So, you know, the doctor even questions you. You know, most of the symptoms of Lyme disease are vague, and without, you know, coming in with a tick in a bag, they're just going to dismiss it, which is a huge problem. So that's why the canine data is so important, because if we can show through canine data that there are actually infected ticks in an area that is generally considered free of infected ticks through the human data, that would be a big step forward. So that's another One Health approach where we really need to use canine animal data in conjunction with human data to get a better picture of what's going on in the environment.
Absolutely. I hope that's implemented.
Well, advocate for it.
Yes.
You know, we have to advocate for it. The CDC needs to include, I mean, the CDC, in terms of surveillance, they look at rabies in animals, and they used to look at West Nile, but other than that, they're not really looking at animal data. The USDA doesn't really do disease surveillance for human use the way CDC does. So there's large gaps that we need to fill using this One Health approach, using animals as sentinels to inform both animal and human health.
Absolutely. Well, thank you so much for doing this. I learned so much, and thank you for being willing to share your wealth of knowledge.
Oh, my pleasure. And thank you so much for your time and attention. And I'm very happy to have been part of this important effort.
Thank you, Dr.
Before we move
Kahn.
on, before we move on, I'm gonna launch our post-participation question, and I'll launch that right now.
Thank you, Dr. Kahn, for a wonderful presentation.
Okay,
Thank you,
terrific.
Alex, for moderating.
Yeah.
That last question or last answer there is actually a good segue, because we'll have more about the CDC surveillance in just a minute here.
Terrific. Take care.
But thank you so much.
Yeah.
Everyone just takes a second to answer the question, and then we'll start to move into our next set of introductions. Does anyone have any general questions while we're sort of transitioning here? Can you post how many CE credits Tick Academy is per day? I will confirm it later, and if you email me about your CE credentials, I can let you know. But I believe we have, oh, perfect. She's able to share the screen. I believe we have 2 CE credits per day for pesticide applicators and 2.5 vector CEUs for California specifically. And to answer Mario, there are not CE credits for Massachusetts. We have Michigan, Oregon, Texas, Washington for pesticide applicators, and California for vector CEUs. Thank you for clarifying.
All right, I'm gonna end this poll in about 15 seconds. So if you haven't answered, please go ahead and answer that question.
And it looks like Dr. Hinckley is here and just about set. How are you doing this morning?
I'm doing great. It's a sunny Colorado day, and I'm happy to join you guys.
It's great. Thank you so much for being here. Josie, I think we can go ahead and end that poll if we haven't already. And it shows the correct answer. There were, I believe, more than one on this one, which I guess is important to note moving forward. Not all of the questions have just one correct answer. You should be able to select more than one if there are more than one correct answers. So thank you for participating in that. Okay. Moving on right on time. Introducing Dr. Alison Hinckley, an epidemiologist for the Centers for Disease Control and Prevention. She holds a BS in environmental engineering from the University of Florida and a PhD in environmental health / epidemiology from Colorado State University. In 2004, Dr. Hinckley began working at the CDC's Division of Vector-Borne Diseases in Fort Collins, Colorado. For 3 years, she worked as an epidemiologist on arboviral diseases, including West Nile virus, lacrosse, encephalitis virus, and yellow fever. For the last 17 years, her area of focus and research has been on the epidemiology of Lyme disease, plague, tularemia, Bartonellosis, and vector-borne relapsing fevers. She also served as principal investigator of the CDC's TickNet program, a collaborative public health network working to foster coordinated surveillance, research, education, and prevention of tick-borne diseases. She currently leads the epidemiology and surveillance team in the bacterial diseases branch at the CDC. Thank you, Dr. Hinckley, for being here. Can we launch the pre-question for this presentation? Perfect. Just take a second and take your best guess. And then Dr. Hinckley, the floor is all yours. If you are just joining us or something like that, we have chat for general questions and a Q&A functions for questions for the speaker. So please be sure to put your questions in there and we'll address them at the end. I think we're good
To get going? All
to
right.
end the poll. Thank you.
And you can see the slides clearly?
Yes, they look good.
Okay, perfect. As Ryan said, I'm excited to talk to you all today. I am talking to you from Fort Collins, Colorado, where most of the Division of Vector-Borne Diseases for CDC exists. And the focus today is on changes to public health surveillance for Lyme disease that happened in 2022. So it's important to know where your data come from and what the limitations of those data are. I'm going to go into that a little bit today. But before I share details on what's currently going on with Lyme disease surveillance, I always feel it's good to start with a primer on what public health surveillance is and why we do it for certain health conditions. So public health surveillance is defined as the systematic, ongoing collection of data that yields information that aids in public health decision-making. So in other words, we're not just collecting data for the sake of collecting data, but in order to inform public health action. Listed here are some of the discrete purposes of public health surveillance: to identify populations at risk, to estimate the magnitude of a health problem, to determine the geographic distribution of a disease, to evaluate the effect of control measures, and to describe natural history of a disease. And so depending on what your question is with the disease, you're going to make certain decisions that help focus in on one or more of these goals. So here's a bit more background on public health surveillance in the United States. The authority to conduct public health surveillance actually rests with the states, not with the federal government. That's part of the 10th Amendment. In other words, CDC doesn't actually conduct any surveillance. The list of specific health conditions to be voluntarily reported from states to CDC and the associated details of what they send to us are set by the governing body called the Council for State and Territorial Epidemiologists, or CSTE. CDC advises CSTE on this process, but we don't vote. So also, it's important to know that all surveillance systems involve trade-offs. No single system can capture everything I mentioned on that last slide. For example, surveillance for rabies, mentioned in the last presentation, is drastically different than surveillance for influenza. You have different goals and you can do different things with it. Another thing to point out is that sometimes passive public health surveillance, it's actually not the best at answering all things related to a given disease. And so some questions are better suited for periodic research studies. So now, specifically turning to human Lyme disease surveillance, CST made Lyme disease a nationally notifiable condition starting in 1991. The primary objectives were to establish the who, when, and where with respect to Lyme disease risk. Over the next few decades, the surveillance system has been pretty successful at accomplishing those objectives. That is, it's clearly demonstrated the patient ages and gender distribution for persons most likely to acquire Lyme disease. You can see some of that in the tables and figures on this slide. These are all from past publications. It also tells us what time of year Lyme disease is most likely to be acquired, where the disease is most common, and where it's expanding. So despite those successes, there have been a number of growing challenges with Lyme disease surveillance, particularly over the 2010s decade. So first, it's actually quite complicated and very resource intensive. The criteria set forth in all of the Lyme disease case definitions that have been in place from 1991 all the way to 2021 required substantial human resources to evaluate laboratory data and to obtain matching clinical information for each person with a positive lab report. So in areas of high incidence, you know that as the upper Midwest, and Northeast, there were an increasingly large volume of laboratory reports to process. In some areas, in some states, tens of thousands of lab reports came in, and trying to match that with clinical information is very challenging. So on top of this, there's limited immediate public health utility for doing it in already high incidence areas. We already know it's a big problem. So in areas where Lyme disease occurs commonly, the problem is well established. It's always present. We know when it's going to happen. And each individual case report does not really influence our public health action. The resources spent processing Lyme disease case reports are thus not available for presentation, education, or surveillance for any other tick-borne diseases. So finally, inconsistent implementation of surveillance case definitions has also been a problem, and variable reporting practices have taken hold as individual states attempted to cope with the sheer reporting burden in different ways. So as I mentioned, by the end of the 2010s, all of this has rendered national surveillance for Lyme disease pretty non-standardized. A prime example of states adopting alternate surveillance approaches to better make use of their limited resources is shown on this map. So this is the US map of reported cases for 2019. States that are colored white are considered low incidence areas for Lyme disease. States that are light gray are considered high incidence, and you can barely make those out as they're mostly covered by the blue dots representing cases. But if everybody looks on there, do you see Massachusetts? I put a red circle around it. This is a state that's absolutely endemic, high incidence for Lyme disease. But in 2016, that health department made the decision to stop actively collecting clinical information for their positive laboratory reports, which meant they didn't have the necessary information to classify cases as being confirmed or probable under the existing case definition. So they reported only a handful of cases to CDC in this year. This is 2019. But their health department staff actually made numerous education and outreach efforts for clinicians and the general public that they hadn't had a chance to do previously. And they weren't the only state that made significant changes to their surveillance practices. It's just the easiest one for us to illustrate on a map. So given the increasing need for resources and the lack of sustainability of the current systems, the states decided to modify surveillance practices. So after multiple years of discussions, I think we started in 2014, a new case definition for Lyme disease was approved by CSTE in 2021. It went into effect in January 1 of 2022. So early in the discussion of possible case definition changes, we recognize that there are a lot of different objectives and goals for public health surveillance in high and low incidence areas. In high incidence areas where risk of Lyme disease is high year after year, case notifications themselves do not trigger any specific public health actions, as I mentioned. There's not an intervention that happens or even an investigation. The goals of surveillance in these areas are really instead to describe the magnitude of the problem, to monitor trends over time, and to inform longer-term education and outreach that helps promote prevention, early diagnosis, and treatment. So in contrast, in low incidence and emerging areas, the objective of public health surveillance for Lyme disease is very different. If there's a report of Lyme disease in a resident of an area without known disease risk, public health surveillance follow-up is critical. It aids in identification of specific areas with risk of Lyme disease, and in turn, works to actively inform prevention, education, and also clinical practice so that more people get recognized if they have the disease in that area. So based on the recognition regarding different objectives, there are key changes with the new case definition. The new case definition reflects separate surveillance approaches between high and low incidence jurisdictions in line with those objectives. Specifically, high incidence jurisdictions report cases to CDC based only on positive laboratory reports. Therefore, they are not conducting or attempting clinical follow-up on any of those positive lab reports anymore as of 2022. This switch improves standardization of reporting practices across high incidence jurisdictions and allows them to preserve public health resources that were being spent counting cases instead on doing other public health actions. So in this case definition, those that have an average incidence of at least 10 confirmed cases per 100,000 population for 3 consecutive years are considered high incidence. Once that's been observed in what's currently a low incidence state, they become a high incidence state and switch surveillance approaches. Low incidence states, however, continue investigating cases fully, and they continue to collect clinical information on all associated positive laboratory reports. The new case definition aimed to expand the laboratory criteria to also reflect new developments, which I'll go into more on the next slide. This case definition instituted a tiered approach to laboratory evidence of infection that didn't exist previously for Lyme disease. And that means specifically the single-tier IgG immunoblot was demoted from confirmatory lab evidence to presumptive lab evidence. It's not recommended for clinical practice by itself. Lastly, this case definition aimed to increase specificity of clinical findings for the probable case definition in low incidence states. So the left side of the slide outlines what constituted laboratory evidence under the case definition previously in effect. This includes culture, which is rarely done for Lyme disease, or positive 2-tier tests for IgM or IgG, and a single IgG immunoblot. And the right side shows the revised laboratory evidence of infection. Under confirmatory laboratory evidence, we have culture, but PCR and IHC have been added, as has discrete mention of Borrelia mayonnae as a cause of Lyme disease in the US. In addition to the 2 standard tier tests, we also now include a modified 2-tier tests, MTTT. This is an approach to serologic testing that was first approved by the FDA in 2019 and includes 2 different sequential immunoassays, both by the same manufacturer and approved in a single algorithm rather than an EIA followed by an immunoblot, which is typically a standard two-tiered test. And here are the actual case classifications. Under the 2022 case definition, high incidence jurisdictions are now using a laboratory-based approach to surveillance. There are no confirmed cases given that lack of clinical follow-up. And in a high incidence state, any reports that meet the confirmatory lab evidence will be reported as probable cases. So two-tier serology and the new addition of PCR and IHC, which we expect to be infrequent. Because the single-tier IgG has been reclassified as presumptive laboratory evidence, in the absence of clinical criteria in high incidence states, they will not be reported to CDC. Now, in low incidence jurisdictions, because the goal is to truly understand if there is local disease risk that will prompt further public health action, labs will continue to be followed up on, and the confirmed and probable case definition are differentiated by the tiered laboratory evidence, although they share the same clinical criteria for both. So above the screen line, that represents all of that which gets reported to CDC. And while there are suspect case definitions, we don't see those at CDC, although they are used in states for some of their information. So to review, high incidence jurisdictions, a probable is a case that meets a confirmatory lab evidence. Suspect is a case that meets presumptive lab evidence. In low incidence jurisdictions, confirmed is clinically compatible that meets confirmatory lab evidence, and a probable is a clinically compatible that meets presumptive lab evidence. So given these changes with the new case definition, we anticipated that we might see once the 2022 case numbers were reported to CDC from the states, we thought there would be new reliance on laboratory positive in the high incidence states accordingly. We expected a pretty large increase, therefore, in the number of cases. Just having cases reported from Massachusetts and some of these other states that were performing alternative surveillance methods again would definitely increase the numbers. However, without erythema migrans reports submitted directly from clinicians in high incidence states, and we knew this happened from time to time, we knew some of the otherwise rock solid cases of Lyme disease under the old case definition would no longer be counted. In addition, some of the less clear, less specific cases would no longer be classified as confirmed or probable in low incidence jurisdictions. So those would likely lower case counts in those states. The big question mark for us was how much the new laboratory test, modified two-tier test, would impact these numbers, particularly in the high incidence states with positive lab reporting only. It's a more sensitive algorithm, and we anticipated that it should pick up more early disease cases than previous algorithms, which is a pretty exciting turn of events for lab testing in Lyme disease. So in the next few slides, I'm going to show you where we ended as of the final report out, which happened in February of this year, 2024, for our 2022 surveillance data for Lyme disease. This bar chart, which I'm sure a number of people have seen some early version of before, is based on Lyme disease surveillance data from 1996. These three colors of this slide represent three major case definitions for Lyme disease. And you can see that between 30,000 and 40,000 confirmed and probable cases of Lyme disease were reported to CDC each year in these recent years, kind of before pandemic. On the far right in yellow, with the 2022 cases under the latest case definition, we see about 62,000 cases. So that represents about a 70% increase over the average of the pre-pandemic years, 2017 to 2019. We did not compare this latest year, 2022 to 2020 and 2021 due to the reporting anomalies associated with the COVID-19 pandemic. So on this slide, we look at data in a lot more depth, and we see that compared to the 2017 to 2019 data, those years I circled on the last slide, the 2022 number is about 1.7 times as high as 62,551. Given the shift to less restrictive criteria in high incidence states and slightly more restrictive criteria in low incidence states, the proportion of cases reported from high incidence areas is actually increased under 2022 case definition to 95.5%. However, the proportion of cases that were male stayed about the same with this new case definition. And just a note, on the bottom of this slide, you can see a reference with Kirsten Kugler as the primary author. And this is an MMWR that goes into a lot more depth on this case definition change. And this was released in February of this year also. So one other consideration, we've historically used illness onset date, and that's obtained through clinical investigation of cases. And we use that to assess seasonality. The data was available, this data was available for about 68% of cases reported during 2017 to 2019, but only about 5% of cases in 2022, given the new changes. So the illness onset peaked at week 26 during 2017 to 2019. You're wondering what week 26 is. I think it's the first week of July. Illness onset also peaked at week 26 in 2022, whereas diagnosis, laboratory test, and reporting dates that collectively peaked about 2 weeks later in week 28. So this figure depicts incidence rates per 100,000 people in 5-year age groups. 5-year age group and sex in both time periods. That's the 2017 to 2019, and then the new 2022. Males here are shown in black lines. The darker one belongs to 2022, and females are in blue. So as mentioned, males accounted for 57% of cases in both time periods. The incidence in 2022 was higher than during 2017 to 2019 for both males and females. The age distribution was bimodal during both time periods, but you can see from this slide here, proportionally more reported cases in 2022 occurred among these older adults, really spiking up here around the 50s into the 70s. And the incidence among children less than 10 years in 2022 was pretty comparable to that reported during 2017 to 2019. And now to wrap it all up. So overall, this new case definition has been a paradigm shift for Lyme disease surveillance for sure. As I shared early on, conducting passive surveillance in the traditional manner wasn't working at all. This new approach is pushing surveillance to achieve objectives relevant to jurisdictions based on their level of Lyme disease endemicity. And ultimately, what this does is free up public health resources from counting cases in already high-risk areas to activities more likely to improve public health. So it's good timing given the potential for a new Lyme disease vaccine on the horizon in the next couple of years. So there were some impacts observed in the 2022 preliminary data, actually final data, that are due to the new surveillance methodology being used. Substantially more Lyme disease cases reported to CDC than before the COVID-19 pandemic. Such cases would have been excluded previously due to missing or inconsistent clinical information or even alternative surveillance practices. The biggest increases in high incidence states were among those that had alternate practices, as I mentioned, like Massachusetts, and the decreases were in low incidence states were due to improved accuracy. Onset dates were rarely available with the 2022 case definition because we no longer received any of that clinical information mentioned previously. And so that information has been replaced with laboratory testing or reporting date. So you'll see seasonality shift about 2 weeks later because of this change in surveillance practice. So there are other considerations. There's a higher incidence that we see among older adults. We think this is in large part due to the increase in lab testing that occurs with increasing age. But also, it may reflect differential clinical practice and stage of illness at detection. There's also an increased potential over time for prior exposure to Lyme disease, as seen with long-lasting seropositivity. What wasn't mentioned in the last presentation is that when people are test positive for Lyme disease, they stay positive often for years to decades. So testing repeatedly would just show evidence of a previous illness or a previous infection. It wouldn't provide a lot of information about new symptoms. So changes in laboratory testing practices in 2022 may have also influenced incidence. The use of the modified two-tier assays increased at commercial labs in 2022 and have much higher sensitivity in early illness than the standard two-tier algorithms. This certainly resulted in proportionally more people with positive laboratory evidence, although at this point we can't say how much. However, health departments have anecdotally reported challenges in receiving or identifying the modified two-tier test assay data within their systems. It's mostly due to the lack of specific test codes to help discriminate between test types. This may have resulted in under-ascertainment of persons with positive laboratory evidence in 2022. The modified two-tier test specific LOINC or lab reporting codes were created and approved in early 2023. So we do feel that there will be improvements to this in the next year of data that CDC receives, which is the 2023 data, should be out in the next few months. So if we revisit the slide from the beginning about surveillance meeting the objectives, the question is, how do we think our new Lyme disease case definition performs? So far, it looks like it'll meet most of our important goals of identifying populations at risk and determining the geographic distribution of disease. Given the improved comparability across space and time compared to our previous definitions, we also believe it'll allow us to evaluate effectiveness of large-scale control measures, perhaps like a vaccine. That'll be particularly important as we see that come online, as I mentioned, in the next few years. And while it does help us to understand the magnitude of the health problem, we know we have a lot of Lyme disease in high incidence areas and a growing problem in some, but not all low incidence areas. It still doesn't count every person diagnosed with Lyme disease. It'll not help us evaluate the natural history of the disease. That information must come from other investigations and research. But ultimately, the new case definition allows for a better use of human resources in the most highly effective areas and makes room for much more work on prevention and education. And that's it. Thank you.
Thank you, Dr. Hinckley, for a great presentation. If we can, do we want to do the poll first and then do the questions? Or launch the poll while we start some questions?
Sounds good.
So again, we have this participation question. No significant pressure here, but please answer it to the best of your ability. Question is from Joellen Lampin, who asks, we have used the reported incidence by 5-year age group and sex as justification for focusing on children. Would this new data suggest that you would shift the emphasis to older age groups?
Well, that's a great question. I think this is one of the cautions of new data. You know, there is certainly an influence of who gets tested for Lyme disease on the data that we now see, because so much of it comes from reported lab testing. We know that children aren't tested as often. They go to their pediatrician. If the clinician believes that what they have is Lyme disease, they're less likely to take a lab test because children are less likely to want that to happen. So they don't get reported as much. Alternately, as people age, we have great data now. You'll see some more come out in publications here soon that shows that, again, as people are getting older, they're getting tested commonly. They're getting tested annually for all kinds of other things. And for that reason, we think there is a bias to receiving positive lab information for older adults that may not reflect what's true about the epidemiology of disease. So we're going to, as I mentioned, get some publications out and do some corrections maybe on this data in the future to reflect the importance, as I said, of children still being one of the highest risk groups, for sure.
Thank you. I saw someone raise their hand. If you are able to put it in the Q&A function, that would be helpful. I don't know. Well, I don't know where they went. But are there other questions? Scott Larson asks, do you think dogs would also test positive for multiple years, as you mentioned that humans would?
I do. The testing in dogs is a C6 testing. It's actually a little bit different than what humans are tested by. It's less specific. So it's a bit more prone to false positivity. I do think antibodies will persist following infection in dogs. And we've seen that in some of the papers, even that were used as references in the previous presentation. That's important when you're looking at the map because you see that dogs, the presence of dogs, seropositive dogs in some of those areas where we haven't seen a lot of Lyme disease could certainly reflect, dogs that moved from other high incidence areas that remained seropositive and were tested annually. Testing of animals and humans for that matter, whenever there isn't an indication of acute disease, increases the likelihood of false positive results. And so some of those maps in the previous slide show, incidence rates for Lyme disease that are really right at what you would expect for a false positive rate. So something to keep in mind whenever you're looking at the maps for animals versus the maps for humans.
Thank you. I think this will be our last question. We're coming up on our break here. It says, do you think it would be helpful to distinguish between BB, SS, and BM? How did the CDC decide to keep them combined versus separate diagnosis?
Yeah, for BM, I assume you mean Borrelia mayonii and not Babesia microti. But so Lyme disease is what we do surveillance for, not for the infecting pathogen. Both of those pathogens result in a Lyme disease infection or a Lyme disease illness that's pretty much indistinguishable from one another. So we're receiving serology results, and the serology test is also, at least for the modified two-tier test, able to pick up both Borrelia burgdorferi and Borrelia mayonii as well. And the test does not distinguish between those, at least unless you use some of those older techniques, the Western blots, where you can see a little bit of a different response.
Great. Thank you so much, Dr. Hinckley. Thank you to all of you for your questions. We are going to take our break now. We will start back up promptly at noon Central Time with a presentation from Rachel Walters from Texas A&M University, Kingsville. So we will be right back. Thank you, everyone.
Rachel, if you're still on, you should have sharing capabilities now for when we come back at noon. Feel free to shoot me a chat, or if you have any questions, I can just go ahead and answer them now.
Okay, thank you. Can you hear me okay? I just want to make sure my volume and everything is good.
It's a little quiet.
Okay, let me see if I can adjust anything in my settings. Is this better?
That's better, yes.
Okay, awesome. And then
Oh, actually, no, it's not better.
I can't
A little
hear.
muffly.
Ah. Okay. I tested all this yesterday and it worked fine.
I can hear you
but-
better when you're a little closer
okay.
to the screen as
Okay.
well.
Maybe I'll just
Yeah.
try to talk louder. Okay.
That's perfect.
Awesome.
That's perfect.
Thank you.
Awesome. Yes, we'll be back at noon. Start with the poll question, and then Ryan will introduce you. So,
Okay.
all right. Thanks, Rachel.
So no problem. Thank you.
As everyone's coming back, I just wanted to take a moment and acknowledge our sponsors once again. So again, at the gold level, we have Tickbox Technology. Oh my goodness, it's going without its mind of its own. Tickbox, Tick Control System, Project Lime, US Biologic, and mainly ticks. At our silver level, we have the North Shore Mosquito Abatement District. And at the bronze level, we have Alexis Chesney, MSND, LAC, and Central Life Sciences. Thank you so much to our sponsors. This would not be possible without your support. And if you would like more information about any of these sponsors, all of their websites are linked on our website. Otherwise, you can feel free to reach out to me and I can give you contact information for them. So thank you.
Oh,
How do I? Did it stop sharing? And we do have another staff member joining us, Nikki Blunier from the IPM Institute will be stepping in to help with some facilitation and monitoring the chat and Q&A once again. Rachel, are you all set?
Yeah, I can go ahead and share the presentation now if that's good with you.
Wonderful. Yeah, go ahead and share your screen and we will launch the poll question. And we'll go ahead and get going. Thank you, everyone. Welcome back from the break. I hope you all enjoyed your time away from your screen. Our next presentation is from Rachel Walters, who is a first-year master's student at Texas A&M University, Kingsville, studying under Doctors Martin and Rideout Hanzak. With interests in zoonotic disease transmission and the impact of environmental factors on disease dynamics. Rachel has studied a range of different disease systems from snail-borne trematodes in rural Pennsylvania to plague in Northern California small mammals. Bringing those passions to Southern Texas, Rachel now studies the impacts of prescribed fires on tick abundance. Following her studies in Kingsville, Rachel hopes to pursue a PhD and career in vector-borne diseases. So welcome, Rachel. Thank you for being here.
Yeah, thank you for having me. So thank you for the introduction. I'm Rachel, and I'll be talking about prescribed fire as a potential tick control method. Before I start, I would like to introduce my research team and everyone who kind of has a role in helping on this project. So Ryan provided a great introduction for me, so I am not going to repeat all of that. But Doctors Rideout Hanzak and Alyn Martin are my co-PIs. They both have experience in disease. Dr. Rideout is a certified senior wildland fire ecologist and a lead burn instructor who has over 20 years of experience leading prescribed fires. So if you have any very specific prescribed fire questions, she's going to be a great contact person for you after this presentation. And then Dr. Alyn Martin has worked on a wide range of disease systems and has current work focusing on parasitism in game species, as well as disease management. And she's working in many diseases as well, including tick-borne diseases and toxoplasmosis. Drs. Scott Henke and Tammy Johnson are committee members on this project. Dr. Scott Henke studies wildlife disease and has a medical and wildlife disease background. He's also focused on a very broad range of parasites, toxins, and zoonoses. And then Dr. Tammy Johnson is a wildlife disease ecologist with Texas A&M AgriLife, and she studies pathogens at the wildlife livestock interface. Not pictured is Dr. Ashley Reeves as well. I wanted to mention her because she is the point person for the project on the East Foundation side, which is the landowner of the ranch that we operate on, and she is a research veterinarian. So I wanted to introduce prescribed fire since I'm not sure what everyone's background is, but prescribed fires are low intensity fires compared to wildfires, and they are used with specific management goals. So this can be to manage vegetation, control pests, or combat woody encroachment. That woody encroachment bit is important for our specific rangeland environment, and I will get into that a little bit later as well. And then some variables associated with prescribed fire that you might hear me mention are burn interval and frequency. That's just how often burns occur and the time between burns. Burn season, this can vary depending on where the burn is taking place and the literature you're looking at. So here in the US, we talk about burn season a lot in terms of winter versus summer or spring or fall. In other countries where they have a more prominent dry and wet season, they may talk about the burns in terms of those seasons. And then burn intensity refers to the scope of the burn, and this can be burn depth, temperature, all of that. And like I said, prescribed fire is often low intensity. I wanted to briefly also go over grassland degradation and mesophication. Grassland degradation in our environment looks a lot like woody encroachment, and it has multiple drivers. This can be overgrazing, land conversion, invasive species, and also altered fire regimes. And like I said, grassland degradation can look different depending on the geographic location. So you'll see a lot of times with degradation, reduced plant and animal species diversity, as well as shrubbification and woody encroachment, where those species will kind of start to outcompete grassland species, and then that will in turn lead to a lower productivity ecosystem. You'll also likely see higher invasive species counts in these degraded grasslands. And then mesophication is a term that applies to our research and is very important. I've seen it more when talking about forested ecosystems, but this was coined by two researchers, I believe, out of the Forest Service. And it's when more mesic species start to replace more xeric species. Mesic species are just more shade tolerant, less conducive to burning. They promote higher moisture environments and create more favorable tick habitat because of increased shade and moisture in those environments, whereas xeric species are more adapted to dry conditions and more tolerant of burning. Texas has many ecoregions, illustrated on this map, and the citation for this map is below. It's a really excellent study that talks about the burn intervals of Texas, as well as the ecoregions if you are interested. But we are in this fourth subregion of Texas, which is circled on the upper left-hand corner. And this is the Gulf Coast Prairies and Marshes ecoregion. The other three maps are three historic burn interval estimates, which all pretty much say that our region of Texas and Texas as a whole has a very frequent short fire interval. So historically, fires returned in the region at an interval of every 0 to 5 years, in some cases a little longer, as you can see on the map. And this basically just means that the native plants of Texas and of our region are adapted to and can withstand frequent fire, and that is healthy for these ecosystems. This excludes some invasive species and also exotic introduced species, which are present on the landscape and on the ranch that we do our work on. And this also means that we can burn frequently for tick control if needed, because the native species are adapted to frequent burning. A little bit of history on burns in the region. So burns were used pretty commonly from the mid-1800s to around the 1940s in our ecoregion. Fires were used about 2 times annually once in the summer, once in the winter, and these kind of petered out, like I said, in the 1940s when more fire exclusion and suppression policies were put into place. And that cessation of fire was cited as a cause for woody plant expansion in the region, and then also potentially to the rise of tick species as well. There are many potential impacts of prescribed burns on ticks. Two main overarching possibilities is that prescribed fires could decrease tick populations, and another one is that they could either increase or maintain tick populations on the landscape. This figure to the right kind of goes over the mechanisms associated with both of these factors, but I will talk about these on the next couple of slides. Prescribed fire can decrease tick populations through direct mortality. So that's direct killing of mostly questing ticks by heat or flaming. And ticks can be resistant to this depending on life stage, species, and their location in the environment. And I'll talk about that in a little bit. Alterations in microenvironment is also cited as one of the major possible ways that prescribed fire can decrease tick populations. Burns can alter the microenvironment in the following ways. They can increase exposure to UV light, reduce humidity and moisture, and also decrease leaf litter. So not only does this remove important habitat for ticks, it also contributes to desiccation. So this is more of like a post-fire impact on tick species. Hosts also temporarily leave the plots during the burns, carrying all the ticks on them with them. So this is a temporary effect of fire that could lead to decreased tick populations. But on the other hand, when you have post-fire regrowth of plants, that's very attractive to certain tick hosts and grazers, and they will likely recolonize that plot in search of food, and then will carry the ticks on them with them if the degree of host density on the plots are very extremely increased by this effect, which is referred to as green bite in some papers. That's just the post-fire regrowth. It could also increase the questing success of ticks, but there has been a rebuttal to this point in the literature where a study cited that shorter grazers may be poorer tick hosts, but this was also, I believe, in a South African savanna environment. So there might be habitat level or species level kind of variables that play into this that we don't have on the ranch. But I did want to mention that there is some pushback against this concept. Another way that tick populations can either be increased or maintained post-fire is through refugia. So soil and leaf litter are going to be the main refugium in the literature. Adults are more resistant to heat exposure in the upper soil regions. And then two separate studies have found survival in 1.2 and 2.5 centimeters of soil. Species also have different tolerances for burning and heat, as do life stages. So this also could depend on what tick populations you have on the land you're managing for. And then in our study, we're specifically interested in looking at moths as refugia, and I will explain moths a little bit further in a second, but basically they're these stands of dense woody vegetation that are interspersed throughout the grassland environment, and they do not burn, which is very important because then the tick species that are present within them do not die from direct mortality or likely alterations in that microclimate. And then hosts will also sometimes use these plots as refugia during burns if they cannot escape the plot. And then the tick species are able to drop off and survive within that moth. So the tick and pathogen species of interest are mainly the Gulf Coast tick, American dog tick, and the cayenne tick for this project. These are just observationally what we have seen on the ranch. We're not excluding the study by any means to these three species, but these are just mainly what we've seen, at least in the adult form on the ranch. Tick Encounter is a tool through the University of Rhode Island, which will report within the past year if there have been sightings of these species in different regions of the US, as well as if they have confirmed transmission. So the Gulf Coast tick and the American dog tick are present on this map, and the Gulf Coast tick can potentially transmit spotted fever, rickettsiosis, and canine hepatozoonosis. The American dog tick can transmit Rocky Mountain spotted fever and tularemia, and has, according to this map. And then the cayenne tick is not on this map, but we have seen it on the ranches, and it's a vector of rickettsia species. These images to the right are from Georgia Southern's database on different tick species, except for this bottom image of the cayenne tick, which is cited below. Another important thing to point out is this name for the cayenne tick. So in the region, we have Amblyomma mixtum, but the cayenne tick is part of this species complex that encompasses, I believe, 6 different species. So if you're hearing the common name cayenne tick, it may be referring to multiple tick species, but in this study, we have Amblyomma mixtum. This has not been particularly tested for yet. We will be identifying the tick species, but that's the one that the literature says is present in this region. So our research questions and study aims that I will be addressing in this talk is, does prescribed fire affect tick density, and what is the role of fire return interval, as well as demonstrating tick phenology in the region through our control plots and characterizing the impacts of burning on tick density. We are also very interested in pathogen carriage as affected by prescribed fire, as well as differences in vegetation structure within the grassland environment. However, we will not be addressing these factors in this talk. We are just going to address those in the future. And I wanted to briefly go over our study site and the burn regimes that we use. So we conduct our research on East Foundation's El Sal's Ranch in Willacy and Kennedy counties of South Texas. And this is once again in that Gulf Coast prairies and marshes ecoregion. And we have 2 replicates of each treatment, so 6 total plots that we conduct tick sampling in. They're marked on the map on the left with the red dots. We have 2 winter short interval plots, 2 winter long interval plots, and 2 control plots. And then to go into more detail about what these plots exactly are, the short interval plots are burned every 3 years. They've been burned every 3 years since 2017, while the long interval plots are burned every 5 years, and they have been burned since 2019. It's also important to point out that these plots are management size, and they range from 360 to 728 acres. And this is quite large compared to other tick and fire studies in the literature. So the factor that these plots are so large in terms of acreage may also have differential effects on the impacts of burn compared to the studies that we have seen in the literature. The vegetation structure is also not homogeneous on the rangeland that we do our research on. So I mentioned mott's before. I did not know what a mott was before I started this study, but it's essentially a sort of cluster of native woody species on our ranch. It's honey mesquite and live oak mostly that serve as refugia, like I said, for both wildlife and ticks on the landscape. And these motts are not bad necessarily. The goal is not to totally remove them. They're very important to wildlife. They provide shade, cover, and nesting, but they're only bad when they start to outcompete and have dominance over the native grass species, and the burns essentially just help to manage and keep this balance. If you're interested in the exact grass species and the forbs species, I've listed them on this slide as well, but this is basically the composition of plants that we see on this ranch. Our field collection methods utilize dragging. We drag 2 750-meter transects per plot, as well as 3 to 4 mott perimeters per plot, and these range from 80 to 418 meters around. These motts were selected randomly, and they do vary on size, so some motts on the ranch are massive and beyond the scope of the project to drag around. Some are only a few trees, so these focus mainly on those mid-sized moths that are the most common on the ranches. And then you can see with this figure that the sampling methods are the same for burn and control plots. So I wanted to go over some preliminary results with you very quickly, and this is the wrong slide, but we will be going over the phenology with control plots as well as the impact of differing burn intervals. And I wanted to point out what actually goes into a point that you'll see on the graph, because it's not necessarily the most self-explanatory when you look at it. And I thought it would be helpful to conceptualize what's actually going into a data point. So for example, if we have adults in control plots, this control plot treatment is composed of 2 plots, which then have 2 transects each and 3 to 4 moths each. So that's around 10 to 12 observations per point, if that makes sense. And I'll have this figure at the next of all our graphs so that you can kind of conceptualize this a little bit better, because there are so many variables that go into this and it is a little bit confusing. The moths and the transects are not separated out at this point. We do have separate data on those things, but for the purposes of these slides, we have them kind of lumped together. Tick species are also lumped together, so that's just important to keep in the back of your head when we're talking about this. So this is phenology per life stage in control plots, and these points are averages. So it's the average tick density per meter squared by life stage across the 2 years of sampling in control plots. We'll break this down by talking about adult versus immature stages, but I wanted to point out briefly that there is a large variation in these densities surrounding the peaks that we'll be seeing, and that the y-axis differs for life stages, as you can see in the graph. But I just wanted to point that out in case it is not obvious. The larvae do have a wider variation in that y-axis compared to the adult and nymphal stages, just because they exist in those clusters, and we do tend to get them in clusters instead of as individuals like we do for nymphs and adults. So this is illustrating the adult phenology component in our control plots. Once again, this is over 2 years of sampling. It's average tick density per meter squared. And we can see that there is sort of a peak in the fall, and that the adults are present at lower densities or even absent in other months, especially the summer months where there is a notable absence. This has also not been statistically evaluated at this point, as with all of the graphs I will be showing you, so that is important to keep in mind as well. And then let me find, okay, there it is. Our immature tick phenology in control plots is illustrated on this graph. I also just wanted to remind you guys again that the y-axis is different for these 2 species, but they do have similar peaks. And we can see that they line up pretty well over the months that they occur in. So they have pretty much their peak in the spring and the summer months of densities, the late spring, early summer. And then I also wanted to point out that these nymphal densities are higher in the early spring of 2023 compared to the larvae. So that was a notable difference between nymphal and larval ticks. And I also just wanted to point out quickly that in the 2023 sampling year, there was more conditions similar to drought, whereas this year we did get a lot of rain. And then this is that same plot, just with raw data in the background for your reference. So those big points, once again, are averages, and those smaller points are the raw data points, just so you can see the variation in the data set. And then the next few graphs I will go over will control those burn versus, we'll talk about those burn versus control plots using raw data to get the averages of density, or using raw data to talk about density instead of averages. Otherwise, the layout is very similar to the previous plots. So this is a comparison of our short interval burn versus control, and you can see that there tends to be the same seasonal variation. So burns do not, at least observationally in this stage, do not seem to be impacting phenology, which has been replicated in other studies. Additionally, the values for density are very, very similar as well. This has not been statistically evaluated again, and we are hypothesizing that this will maybe change when we separate out moths versus trans ticks, because remember that moths can act as refugia, at least that's our hypothesis, that they will act as refugia for ticks, and that high density of ticks in moths can potentially override the effect of fire, or at least looking in the data can limit the effect of fire seen in the transects. But once again, this is hypothesized at the moment. Compared to control, we also, or compared to long interval plots, we also see a very similar trend in both density and phenology among the plots. I wanted to point out for the long interval plots that although they've been burned since 2019, tick counts were not started until 2024. So that is why you see this missing year for tick collection in 2023 among the long interval plots. And then this is just a look at everything together. Observationally, there are similar raw tick densities across treatment types, as well as differing phenology among life stages. And then those are pretty much our conclusions, at least for this slideshow. But I did want to point out a few key points. So talking about phenology, seasonality, and the differences between seasonal activity between immatures and adults. This can largely be expected with ticks. We do see seasonal variation. However, because our climate is quite different down here compared to other regions of the US, we didn't want to take phenology for granted, so that's why we are demonstrating it. We do not normally have very large freezing events. We can sometimes have drought, and we have high temperatures that are sustained for most of the year. So it was important for us to illustrate phenology in this study. And then observationally, we have seen similar tick densities with control plots compared to burn plots. But like I said, we hypothesize that vegetation structure may play a role in this similarity, and we will be separating those points out in the future to look further at that. Future directions include looking at that difference in vegetation structure, as well as conducting statistical analyses. And we also are going to identify ticks to the species level using both morphological and molecular methods. And then we also are hoping to test for a variety of pathogens, including Ehrlichia, Rickettsia, Anaplasma species, as well as tularemia and hepatozoonosis due to the ticks vectoring potential of those pathogens. And I also just wanted to point out before I end this presentation that burns are a disturbance, even if they are low intensity. Burning depends on many factors. So if you're looking to burn for the sole purpose of tick management, it might not be a good idea if you're ignoring all of the potential impacts of burn on the ecosystem, because it is not a specific treatment for just pests. However, if the environment that you manage could benefit from burns or is already adapted to burning, they could be an important land management practice, as well as potentially controlling for pests. And then if you have any questions, we can take some now, but you can also email either me or my co-PIs. And then if you also just Google anyone's name, we have website pages as well, and Dr. Rideout has a very amazing podcast if you are interested.
Thank you so much, Rachel. I think we have a few minutes for a couple questions. Can we please launch our post presentation quiz? And we'll start with the first question. It says, could you please explain why there are negative tick densities in these graphs.
Yes, so the negative tick densities are a result of the standard deviations. If you want me to go over the graphs in more depth or send you those images, I will gladly do that. And you could email me or my co-PIs afterwards. That was actually a question I just had for my advisor as well. And it is a result of those standard deviations.
Great. And this one says, would you expect to see differences in biodiversity, plant and animal, between plots over time?
I would expect to see some differences, especially with plant communities. Observationally, the animals present on the ranch seem to be in relatively equal densities around the plots. They have pretty much free range of the ranch as a whole. There are some gates, but they are relatively low, so we do see pretty similar animal distributions across plots. For biodiversity, I would expect to see differences in some plant composition all the time in this or throughout time. And this has been seen just observationally in our plots. Some will have really, really high densities of Gulf cordgrass. Others will have really, really high densities of forbs. And overall, they do have the same plant composition, but they are present at different densities.
That's great. This might be our last question. Does that mean that climate zones are pretty similar between plots?
Yes, they should honestly be the same. The ranch is very massive, so there might be slight variation, but they should be similar, yes.
That's great. We are right about at time. But Rachel, I want to thank you for this presentation. And
Yeah,
thank you all
for having me.
for your questions. We'll transition to our next segment here. Sorry, I have to change some permissions. Rachel, are you able to stop sharing?
Yes, sorry, I didn't know I was
Perfect.
still sharing. Let me, oh, it does say that I'm not, wait, no, okay, never mind. I found out how to do it. Okay. Okay, is that good?
It's good. Thank you.
Okay, no problem.
Okay, moving into our next segment here, we have a sort of small panel discussion. We're going to talk about emerging tick control technologies with David Whitman and Jolieke van Oosterwijk. So David Whitman is the president and owner of TickBox Technology Corporation, which manufactures and distributes the TickBox tick control system and its two-piece metal protective cover in Norwalk, Connecticut. And all of its components are made in the USA. We'll have a little more information on it in a second here. And Dr. Van Oosterwijk is the Chief Scientific Officer at US Biologic, where she leads all development of US Biologic's Auris Bio platform, including orally delivered antimicrobials and vaccines for animals and humans. Most recently, she is leading development of a novel chewable vaccine to reduce pandemic influenza. So the way this is going to work, is we have, we're going to get a brief overview from both of our presenters here, and any questions you have for them about their work or their products or emerging tick control in general, please feel free to put in the Q&A, and we will get through as many questions as we possibly can in the next 25 minutes. So the first thing, it looks like both of your videos are working, but if you can both introduce yourselves and give a background on your organization's tick control products or methods, just a little more than what I just went through.
Yes, I think I'll go first. Thank you for the introduction. So yeah, so Chief Scientific Officer at US Biologic. We're in Memphis, Tennessee. We are very much focused on this One Health strategy, as everyone heard about at 10 this morning from Laura Kahn. We are looking at all these tick-borne diseases and looking like, what are these reservoirs and how can we stop the spread at the reservoirs? So for that, we have developed a station, just as my co-panelist. Our station does a little bit different. So what we have done is we are actually in the vaccine business. So we make orally delivered vaccines and therapeutics. Because you can't go around the forest hunting down mice and injecting all of them. I mean, you could, it's a lot of work, it's not effective. So we thought, like, if we want to vaccinate mice, and specifically with our first product that got conditional approval from the USDA this year, is a oral vaccine for Lyme disease, so that it has to be oral because the mice still have to eat it voluntarily. And then it has to be completely stable at different temperatures, room temperature, hot summers, cold winters. And then we made a station so that it would be automatically opened every 2 weeks. So the pest management professionals wouldn't have to go out every 2 weeks to do the booster for the mice. Also because these mice, they might get eaten by predators, or they just die because of old age, or because they have new litters. So there's new mice coming into these habitats all the time. So we are giving them their boosters or their first dose automatically. All of this is based on years and years of research, of course. And then we developed that chemistry around it as well to make sure that it's orally available. So our first product is then a Lyme vaccine given to mice to stop the spread of Lyme disease before it gets to the tick. And then we're working further on other vaccines, such as anti-tick vaccines, etc. Over to you, David.
David, you are muted.
Thank you, Ryan, and thanks for having me here today. It's a pleasure to be here. And can you hear me now?
Yes, you sound great.
Oh, great. So my brother, my partner, and I have been in the tick control business since the late 90s. We happen to be the manufacturer and distributor of the TickBox tick control system, which was developed by the Centers for Disease Control and Prevention. And now we are the manufacturer. We also own 5 tick control companies: Connecticut, New York, New Jersey, Rhode Island, and Pennsylvania tick control. So we do work in 5 states, and basically that's what we do. And we're here in Norwalk, Connecticut, produced here in Connecticut as well. All the components of our product are made in the US, and very effective product.
David, can you explain a little bit how your product works?
So the tick box is installed preemptively to the emerging nymph populations, and then the second set for in advance of the larval emerging population. So chipmunks and mice will enter the box to eat a non-toxic bait, and at that time they are administered a dose of fipronil solution, one of the best insecticides ever made. And any tick that is on that rodent will die. There could be as many as 400 ticks on a mouse or a chipmunk. Those ticks will die, and then any new tick that attaches to feed will also die. By the second year of treatment, we get over 97% control, 88% control in year one. It doesn't hurt the rodents. It doesn't increase rodent populations, but we get a beautiful control without any additional pesticides introduced into the environment. It can be a standalone product. And it's something that's done indefinitely. But it's great on homes with property, with children and pets and all that.
Thank you, David. It doesn't look like we have any questions yet, but I do want to reiterate that this is an open forum, and if anyone has questions for either of our panelists here, please put them in the Q&A question, or in the Q&A the Q&A area. Both of them are very involved in tick control and have a wide breadth of knowledge here. So any tick control questions you have?
So
First one. Oh, sorry.
I'll have a question.
Go
I'll
ahead.
have a question. So your tick control system, as you say, is a standalone system. And I think when looking into your company, you sell and you place these systems yourself. Does that, are you open? Like, how do customers feel about that? Like, what is with customers that, for example, have rodenticide stations already in their yard? Like, does that interfere with your technology?
Well, we offer a number of programs, one of which can be a standalone product, the tick box tick control system, for people who don't want pesticides introduced into the environment. But we also sell an integrated tick control program, which, in addition to the bait boxes, we'll offer a reduced number of knockdown sprays in year one. Because even though we get 88% control by the end of year one, these ticks are either going to be on the rodent, you or your pets in year one. So that's why we like to introduce a knockdown spray in year one. Year two, you can minimize those sprays, and by year three, the tick boxes are doing all the work. But we do install these tick boxes based on the study and science of one box every 30 feet to treat the chipmunks and mice within their ranges on such property. I don't know if I answered your question or not. But...
No, I was indeed looking for that integrated tick management statement, because I think that's what a lot of us are working towards, right? Like, especially in the beginning, to kind of like get that control. So because like that's what we're also very much looking for. Like, it's standalone, but you can also use it in conjunction with other products.
That's great. Thank you, David. I have, I think, a question for Jolieke here. It says, can you elaborate on the universal tick vaccine and prospects on timeline?
So it's always very difficult to elaborate on timelines when you're talking regulatory. What I mean when I'm talking about universal tick vaccine, we're working on a technology that inhibits the tick feeding on the mouse. So it's where the mice develops antibodies that will respond to the tick when it's feeding on it. So a biological method to repel the ticks from the mice. So it would be the exact same system that we're doing now, but instead of having the mice develop antibodies against the disease specifically, now we're making it develop antibodies against the ticks.
Thank you. Question for both of you. Do either of you have concerns that your baits alter mice and rodent movement that would alter tick distribution?
So for us, we have specifically used completely non-nutritive pellets. So the mice still have to go looking for food. So it doesn't alter the ecosystem or their foraging behavior. And there are no attractants. So it's the mice that just wander around anyway, and it's their normal foraging behavior that leads them to find the pellets.
The bait boxes are installed one every 30 feet. This takes care of the different resident population families within their range. So they don't need to go far for food. And it's right in the ecotone area where these rodents are going to find their nuts and worms and roots and all. So there hasn't been any evidence that they rely on this alone. They eat the bait and then they go back to whatever else is in the area to feed upon. And so one every 30 feet in qualified areas, and they're getting, they don't have to go far.
Thank you. I think you guys just answered a couple questions at once, so try to find. Here's one for David is, can a homeowner deploy their own tick box system? I was under the impression that a certified applicator had to be the one to place these in the environment.
The tick box tick control system is registered in 26 states and is available from professional pest control operators and lawn care companies who have been trained in the use of the product. It is not a homeowner product, and so that should answer the question, I think.
Thank you.
So for our product, so we are currently, so we have USDA approval. We are currently selling in 19 states and District of Columbia. I always have to say that like that. So so mainly we've focused on the Northeast currently, and we are selling it through the pest management professional. So they will be installing the stations, and that is going back. Same with the tick stations, like they have to be installed in those ecotones, so to make sure that they're being installed exactly where the mice and the ticks are living. So your pest management professional can install them, so, and you can go to our website if you don't know who to contact. We can get you in touch with a pest management professional in your area.
Thank you. I think this one could be for either of you. It says, when you say response to the tick bite, do you mean something in the tick's saliva as to create a premature detachment? So
Yeah. So we're targeting saliva proteins. And we're
David,
actually like, so it's not as simple as just this. I'm assuming this is an academic, and I'm willing to talk about this at length with the person asking all these questions. It's not as simple as just simple detachment. We're actually completely interfering with the uptake and the metabolism of the tick.
yeah, I do think a large portion of our audience is academic and
Yeah.
interested in those
Mhm.
those details as well.
Mhm.
So if you'd like to elaborate, you're welcome to.
Yeah, so what we're specifically targeting is the water homeostasis in the tick, so that like when the tick is feeding on the blood meal, like it takes up blood, spits back all the sera that it doesn't want to get more and get all these nutrients. We're targeting it so that the tick gets confused and thinks that it's full and has gotten all the nutrients, but actually it hasn't. So it swells up just like normal, thinks it has completed its blood meal, but it has not had all of these nutrients because of these the channels that it needs for the sensing. Those are now blocked by the antibodies that are produced in the mice. So now the tick thinks it's ready to go to its next life cycle, but it's not, and that's where it dies. So that's where we're doing population control.
Thank you. Another question, is the insecticide in the tick box applied through a spray?
The fipronil solution is impregnated into a wick, so the rodent will pass through the box, rub against the wick, administer the dose of fipronil solution. That actual one-time treatment will kill ticks for 42 days. However, the rodent is in the box every day. So basically you're getting 180 days of tick control. 2 sets of boxes, 90 days each.
Great.
It's good. Go ahead.
Oh, no, sorry, if you have more,
I
go.
just was thinking that it's completely safe, it's childproof. Nothing get in the box except a rodent. Pets can't get to it, children can't get to it. Nothing is leached into the environment. So it's set it and forget it. It's all inside the box, and you're getting tick control without any pesticides into the environment if that's the way the client wants to go.
Thank you. I think we've answered the first part of this, but when using food bait for either vaccine or pesticide dispersal, it seems it would impact population size and or behavior. David mentioned this wasn't a concern. Has this been studied for each technology?
Dr. Mark Dolan, formerly of the CDC, made a publication, it's in our brochure, that there's no evidence of increased populations by treating the animals and luring them in with a non-toxic bait.
Yeah, I think similarly for us, like we've done over the past 15 years, there have been many field trials done with this. We've done a specific field trial where we looked at specifically mice eating them. This was in Connecticut, where we looked at the mice and like used rhodamin B as a biomarker. So if the mice ate it and metabolized the rhodamin B, then we had banding patterns on the whiskers of the mice. And with that, we could also do counts to make sure, like as a secondary outcome. And in none of these studies have we ever observed any increase in the numbers of mice. And as I said, we have been particularly careful in making sure that there is absolutely no nutritive value to the pellets that we're putting out.
Thank you. Two sort of similar questions about the range of products. David has said that the boxes get placed every 30 feet. But what is the range of area for these two treatment options, and what scale do you believe they could be deployed? Could these be deployed? The example given is, would this work for a public park?
Yeah, so we have
Go
one
ahead.
station for half an acre. And yes, this can be used in a public park. This can be used in forests, along trails, golf courses. We have two mechanisms. So the stations, which are mainly intended for around the home use, because of the same concerns that David mentioned, like you might want to make sure your kids, and not that there is any harm, but just for a feeling, right? For homeowners, that nothing else can get in it other than the mice. But like for public parks, there's also broadcast method. So that's where these pellets can just be distributed in the wild.
The tick box host-targeted approach is ideal for schools, daycare centers, nursery schools, camps, and parks, because most states prohibit the use and application of any pesticides that actually do work. So this product is embraced by states to allow treatment to protect children in these facilities.
That's great. Yeah. Next question, and we'll try to get a few more in here. Both of your products focus on mice, which we've talked about a little bit, but is there plans to expand the products for larger potential hosts such as deer? Maybe what would that look like? Has it been studied on your end?
So the The tick, what was the question again? I'm sorry.
Starting with confirming that both of your products are focused on mice, and then have you
Okay,
expanded
gotcha.
products to larger
Right.
hosts such as
So the
deer?
tick box, bait box technology, it treats the competent reservoirs animals of the Borrelia burgdorferi. So we're talking about chipmunks, mice, moles, voles, and shrews. These are the competent reservoirs, and that's what's targeted and has been effective for quite some time. As far as deer are concerned, deer and the ticks they drop are a moot point when you utilize the tick box tick control system, because any tick that's dropping off a deer has fed already, and they only feed once in each stage. So the real concern is the future offspring of those adult ticks, which are going to be dealt with as larvae and nymphs when they feed upon the rodents. So when you use this product, you don't have to worry about the deer.
Thank you.
Yeah, so we're not looking into the deer for Lyme specifically, but we will be looking into products for larger animals such as pets, for example, for the Lyme vaccine, because the oral vaccine, the beauty of it is that you could just administer it at home, right? So it would be for pet owners ideal. So we are thinking about those things for once we get into the stage with anti-tick vaccine, then larger animals such as deer and cattle do become interesting. So those are definitely on our radar.
Thank you. There's a question about academic papers where they can read more about vaccine development and product support. It looks like Jolieke is typing an answer.
Yeah, so we have a
that.
lot of academic. Yes, I'll put those in. There's a lot of academic papers that we published on this. So that there are publications. So, and remember, we are on the market already. So the data is out there for you to see.
I think David mentioned a couple as well, along the lines of population control. There's also a question about where to find more information. So I will link both of your websites in the chat. Let's see if we can get in.
So the tick box tick control system is published in the Journal of Medical Entomology, and that is available on our website for downloading. It's also important to know that this product treats both chipmunks and mice, as another product on the market doesn't treat chipmunks. So if you ask me, we believe 50% of the Lyme disease equation is left out. So we feel the tick box is the best way. The best tick is a dead tick.
There are all kinds of questions. We've talked about products being safe for humans and children and things like that, but has there been the question is, what if a mouse was ingested by a cat or other predator? Would there be any effect that way?
Well, there's a mountain of research on, of course, we
No,
applied to
no,
the US EPA.
For vaccines, it won't be like we have antibodies when we get infected, right? It's the same effect. The vaccine gets broken down in the digestive system. The mouse will make antibodies. Sorry.
And David?
Repeat the question, please.
The effect of
Oh,
ingestion of treated mice by
Yeah.
other animals.
So, of course, there's a mountain of studies and research that's given to the US EPA before registration for approval. They study red-tailed hawks, barn owls, all animals and amphibians, etc. It's published that a cat would have to eat over 300,000 mice in one day to get an ill effect. So there are no issues with the wildlife. Safe.
Thank you. Let's get maybe one more. There's a question that says, have you had interest from any national parks to blanket a highly hiked area? For both of you, David, if you want to answer first.
The answer to that is no, but we do sell to the city of New York. They're using it in their park, specifically Staten Island. So we are selling to them, and there's some other projects in the work internationally. But basically, it's the residential and public and private schools, not so much that.
Yeah, so we have been working with a number of public parks, and we're in the works of designing projects specifically for a park to get those projects off the ground in 25 or 26 to get those parks covered. So there's interest.
Wonderful. I guess we do still have a minute. Are both products targeting Lyme disease or all of the tick-borne diseases? I'm assuming all of them because they're sort of a catch-all, but what does research look like on the back end in terms of tick-borne diseases?
So our vaccine currently is just for Lyme disease, and we're developing more products to cover other tick-borne diseases. I think your answer is going to be different.
Well, any disease that a black-legged tick could transmit is not going to happen if we're killing them. And with over 97% control of the questing tick population on a treated property by the end of year 2, we've also reduced the infection rate in that remaining tick population to less than 2%. So quite effective.
Well, I want to thank you two both very much for your time. If you and both of their websites are linked in the chat, so please go visit them. And both David and Jolieke are representing Tick Academy sponsors who have been very generous with their time and support for this event. So thank you very much for being here and providing your answers. Thank you to all of you who asked questions. Obviously, we didn't quite get to all of them. And if you liked this format, we have another panel discussion at the same time tomorrow, and that will be on mosquito control resources that could be useful for tick control. So thank you, David and Jolieke. And we'll be transitioning to our next presentation in just a second here. Thank you.
Thank you.
Our next presentation will be Dr. Ayra Sayyed, who I think is here, and I hope I said your name correctly. Please correct me if not. I work to get you ready to go here. Hi, good afternoon. How are you?
I'm doing well. How are you?
Good. I can hear you great. Are you all set to start sharing your presentation?
Yes.
While you're getting ready, Josie or Nikki, one of you share the pre-question. Thank you.
Are you able to see my screen?
It says you are screen sharing, but I don't see. It's just a black screen at the moment.
Um,
It says double click to enter full screen mode. I think that's for participants. Yeah. Can you maybe try sharing one more time? Stop sharing and
okay,
reshare. Yeah.
Let me retry that.
There it is.
Got it?
Yep, it looks good.
All right.
All right, Dr. Ayra Sayyed graduated from the Massachusetts College of Pharmacy and Health Sciences in Worcester, Massachusetts. She then completed the R.J. Apel and C. Nelson residency and fellowship in the optometric rehabilitation at the Padula Institute of Vision in Guilford, Connecticut. In 2021, Dr. Sayyed earned her fellowship from the American Academy of Optometry. Excuse me. She is currently finishing her fellowship with the Neuro-Optometric Rehabilitation Association. She's conducted research and published multiple articles in medical journals. Additionally, she has presented at various optometric, rehabilitative, and medical organizations. Dr. Sayyed is the coordinator for externships and residencies at the Padula Institute. She serves as a research coordinator for the Padula Rehabilitation Technologies, where she helps develop and refine new optometric rehabilitative technologies. She has worked as a medical consultant for SphereGen Technologies in New Haven, focusing on the development of mixed reality and virtual reality diagnostic and rehabilitative devices. Additionally, Dr. Sayyed is a medical consultant for NeuroAI. So take it away. Thank you very much for being here. That's
Thank you so
all
much.
yours.
Thank you, Ryan. And thank you, everyone, from the organization to invite me for speaking to you all today. As Ryan mentioned, I am Ayra Sayyed. I am an optometrist by training, and I have specialized in neuro-optometric rehabilitation. And over the last few years, we've been working with tick-borne infections more and more. So our institute, the Padula Institute of Vision Rehabilitation, is located in Guilford, Connecticut, and right now we are seeing patients not just from the Northeast, but also all over the U.S., even internationally, who are coming in specifically due to visual dysfunction that they're experiencing due to tick-borne diseases and complex neurological conditions and findings. So my conversation today will be about focusing on visual dysfunction that is associated with tick-borne infections, and just visual biomarkers that can help with diagnosis, detection, and even management or rehabilitation of these findings. So I'm sure many of you are familiar with and have seen or heard of patients with tick-borne infections. So the variety of patients that are coming into our office are going to be patients who are more chronic or have a neurological condition. And one thing that we hear frequently between me and my boss or the other doctor in the practice is that often the initial symptoms for these patients were something along the lines of a visual symptom or a visual finding. And these were often preceded by other signs and symptoms, or along with other signs and symptoms such as pain and fatigue, fever, things like that. The issue occurs when these patients are going into their local optometrist or ophthalmologist. They are often being diagnosed with binocular vision anomalies such as convergence insufficiency, accommodative issues, reading difficulties. And due to these findings, they end up being diagnosed with a binocular vision dysfunction and then often treated with either lenses, prisms, or even vision therapy. The problem occurs because doctors are not necessarily focusing on the underlying cause. They're just treating the symptoms instead of looking after what could potentially be causing these symptoms. And that is unfortunately due to a lack of understanding of what tick-borne infections can cause for the visual system, but specifically how the brain processing vision. And unfortunately, all of this leads to delayed diagnosis or misdiagnosis, and many of our patients continue suffering, and the worse their conditions are getting over time. The most common signs and symptoms that many of my patients, or generally tick-borne patients with tick-borne infections, are going to report are going to be inflammatory conditions. So in the acute care setting, it's going to be conjunctivitis, iritis, uveitis, basically inflammation of different parts of the eye. Bell's palsy is another one, which is cranial nerve 7 related palsy. That's going to be more of an acute finding. But I have to say that most of our patients, since they're more chronic or more neurologic, I see more of these characteristics of blurry vision, light sensitivity, visual fatigue, visual static, snow, double vision, a lot of difficulty with reading, scrolling the screen, cognitive difficulties as well. And another really common one is going to be how they're feeling in busy, crowded environments. And often our patients are going to say that they get extremely overwhelmed or feel anxious, might even have panic attacks when they are in an environment like that, which is very classic of what a post-trauma vision syndrome patient may present with. And that is all occurring due to a compromise in normal visual processing, which is also a common finding in a patient with tick-borne infection. So before we get further into it, I do want to talk a little bit more about research and what the two visual processes are and how they work. So I have to give credit to the research of the early 20th century scientists by the name of Leibowitz and Posts, who first found the bimodal visual process, which is basically the foundation of what we do in our office and what many of neuro-optometrists are using and doing. So the bimodal visual process is the two processes that we all have that should be working in good, proper balance. The first system is the focal system, which is a non-gravity specific system. It is a detail or attention or acuity driven system that is responsible for concentration identification. So it is present to now. So it is a reactive system. It's a conscious system. Now this system is related to our parvocellular cells, and it is a slower processing system. The second system is our spatial system, which is a gravity-specific system, which is closely correlated to posture, balance, movement. It is a proactive system, so it's a pre-conscious system that is constantly anticipating changes. Now, this system is related to the magnocellular processes, so it's a faster processing system. The spatial system is the foundation of everything that we do, because the focal system is more of a higher cortical level process. So these two systems have to be working in as perfect balance as we can possibly get them. But if there is a compromise in the spatial process due to neurological injury or some chronic condition, the spatial system is first affected, leading to overfocalization and using of just one of the systems. So this is going to be a very important part of what we are seeing with many of the individuals coming in to see us in our office. So again, just the point that I'm trying to highlight here is the balance in the two system is extremely important for normal functioning. And whenever there is an imbalance in normal processes that causes visual dysfunction, Lyme patients are presenting with this exactly, is that the pre-conscious or the spatial process is not working as well as it should. So it is a system that once it's affected, the focusing system or the focal system takes over and then starts creating these signs and symptoms of post-trauma vision syndrome, where many of the symptoms are going to be light sensitivity, headaches, visual snow, visual static, difficulty with posture, balance, and movement, because the lack of organization from the spatial system is not there to provide proper feedback and feed forward processes within the normal processing of vision. So it is also important to just highlight some basics of neurology without getting into too much detail here. But the spatial system, when we are talking about it, we really cannot ignore what's going on within the midbrain. So I'm just going to highlight the part of the brain that we are focusing on when we're talking about the spatial visual process. It's going to be the midbrain specifically, but even more in detail is going to be the superior colliculus, which is just a little bump right on the back of the midbrain. So our sensory information from our eyes traveling through the different pathways goes within the midbrain and registers in the superior colliculus. But also the motor system or the motor input from the body travels up the spine, up the spinal tectal tract, and then registers within the midbrain, the superior colliculus specifically. And then it gets feed forwarded to different parts of the brain. And then the responses are sent to pretty much everything in the eyes and the body. So everything listed here, the different lobes, the sympathetic, parasympathetic nervous systems, cranial nerve 3, 4, and even what I have not listed here, 5 and 7 as well, because all of those can cause significant visual findings causing to oculomotor dysfunction. So pretty much everything that we are organizing and using with our vision can be affected by an imbalance in normal processing within the midbrain. The compromise of neurotransmitter can also occur due to a compromise of the spatial visual process. So the two neurotransmitters that are the ratio between them are required for proper balancing. So the ratio between the GABA and the glutamate principal cells, they both play a principal role in maintaining the balance between excitation and inhibition of the visual system. So if there are imbalance, they can cause characteristics of visual dysfunction. So GABA specifically is used for contrast sensitivity, fine-tuning of the visual input through lateral inhibition. And glutamate neurons are going to be more responsible for complex visual processing, such as detecting motion, color, and depth. So once the imbalance starts occurring in the midbrain, everything else, as far as even neurotransmitters, the autonomic nervous system, the cranial nerves, everything will be affected. With the autonomic nervous system, we all know that it's not an unusual finding for an individual for tick-borne infections to have a dysfunction of the autonomic nervous system. So both the parasympathetic and sympathetic nervous system has a lot of input within the eyes and how the brain process is working to control eye movements and processing vision in general. So any imbalance that is occurring systemically or neurologically, it's also going to affect the eyes and how the individual is processing vision. So some of the most common findings here are going to be pain around eyes, tearing, light sensitivity, headache, neck pain, occipital neuralgias. But it can also lead to neuropsychiatric symptoms, and vision is a really important component of individuals with PANS or PANDAS, or even having OCD or other panic disorders or things like that. So over and over again, the superior colliculus is going to be a very important component when we are going after the spatial visual process and how the visual processing dysfunction starts or occurs. And before we go further into this conversation, I'd like to also talk a little bit more about post-trauma vision syndrome, because I've mentioned it already a couple of times. When I talk about post-trauma vision syndrome, I don't just mean what's going on with TBIs, concussions, head injuries. Post-trauma vision syndrome, I'm going to use as more of an umbrella term that is linked to or correlated with any imbalance in normal processes. And that's how we actually started working with tick-borne infections, is because many of our patients were initially coming in with traumatic brain injuries or concussions that just were not getting better until we realized that the underlying component was an actual active tick-borne infections. And once we started treating some of those, we started seeing changes and improvements relatively quickly. So post-trauma vision syndrome, this research was first published in 1994, which is when it was discovered by my boss, Dr. William Pagela. He was looking at traumatic brain injury patients, patients with visual symptoms, and they used a diagnostic tool called visual evoked potentials, and they looked at what is going on within the patterns of the VEP. And I'll go more into detail with this. So they did pre and post treatment with prisms or therapeutic prism lenses, and they noticed that there was a significant change in using prisms to rehabilitate or treat the brain process. And they were able to create significant changes in how the spatial visual process works. So by using what's called binasal occlusion and basin prisms, we were able to create a change in the VEP amplitude. So you can see in the pre-prism, it's a compressed amplitude versus a treated with prisms, there's an increase. And I know those images are very small, but I'll go into more detail with that a little bit later. So similar to tick-borne infections, post-trauma vision syndrome patients are also very similar here. There's going to be a lot of overlap of findings here of blurred vision, light sensitivity, double vision, headaches, hallucination, difficulty to tolerate busy, crowded environments. And again, it's exactly the overlap that led us to starting to work with patients with tick-borne infections. And all of that occurs due to an imbalance in how normal brain processing is working, leading to a spatial processing dysfunction. Now, another significant finding that occurs in this population is a visual midline shift syndrome. And this can occur due to a disruption, again, in normal processing, causing a deviation in the center of mass, leading to poor balance, difficulties with standing, walking, leading to risk of injuries, risk of falls. And a visual midline can be shifted right, left, anterior, posterior. So think of it as just as a compression or expansion of space is what a visual midline shift is. So the easiest way to demonstrate this would be, let's say if I was holding on to a balloon in both of my hands and I were to compress one side, the other side would expand and one side would compress. So that's essentially what a visual midline shift is, but an individual is going to be doing that with their posture. So they're going to orient themselves in space based on what their visual system is providing them. I do have some examples of this coming up. How we treat visual midline shift syndrome is by using therapeutic prism lenses, because prism lenses do exactly that, is they cause expansion and compression of space. So I do have a video clip here that I just want to share with you really quick of a patient that came into our office. She was diagnosed with just having seizures and muscle spasms, but this is how she was walking when she first came into our office. And one of her symptoms was that her vision she was always in a state of tunnel vision, and that would cause her a lot of difficulties with walking because she wasn't seeing her space. So she would frequently run into objects, walk into objects, or even with driving, she was unable to drive due to how unsafe it was for her. Now, within a few minutes of using prisms, we were able to create this much of a change with her. Now she just has some prisms on, and I won't have time to share the whole video clip here, but once she starts experiencing them, she's even telling us that how her space completely opens up. She starts feeling more and more grounded, where she actually knows where she is in space, and she's more connected. She knows where her feet are as she's walking around the room, and she's actually taking in, processing her whole room. And we're able to create this much of a change in the brain process within minutes. So the goal to treatment with PRISM is to start with something maybe a little bit more aggressive and then slowly back the patient down. So we are rehabilitating the system rather than getting them used to the prisms all the time. So next, I just want to share some research here of some of the biomarkers that I had mentioned earlier. And you can use the QR code if you wanted the link to this, this study that we published last year in the International Journal of Infectious Diseases. So as I have mentioned, mostly we used to work with traumatic brain injury patients, but many of our patients that were coming in, they continued having symptoms, and they were extremely severe. And we weren't able to create any changes until we started looking for the underlying cause. And this is where tick-borne infections really came into our practice. So for a while, between the other doctor and myself in our practice, we were tracking about 20 patients that were having certain biomarkers. So the biomarker of peripapillary ischemia and the visual evoked potential N75 biomarker as well. And out of these 20 patients that we tracked, we were suspicious of tick-borne infections or co-infections. So we discussed the findings and we gave them IGeneX testing kits. And to our surprise, 18 of those 20 patients actually came back positive for either Lyme or one of the other co-infections. So that led us to writing this paper and publishing it last year. So 18 out of 20 patients who were positive, so that ended up being a higher sensitivity than what the current gold standard in testing is with ELISA and Western blot. So what peripapillary ischemia is, we're looking at images right here, which are images of the retina up here of the healthy optic nerve. And down here is an OCT scan looking at the blood vessels in the deeper layer, so the retinal peripapillary capillary region. And this is the optic nerve in the right and the left eye, and then the rest of the retina, the macula is here, which is responsible for the 20/20 part of your vision. Now, these are images of an individual with tick-borne infections that we saw. So we're looking at the optic nerve. This is the right eye, left eye. We're seeing that the rest of the retina is orange in color. There is a nevus here, right here. But we are specifically looking at what is going on. So this graying or ischemic pattern of right around the optic nerve head in both eyes. And then when we followed that up by scanning the deeper layers of the retina, we also noticed that there is a loss of blood vessels in both eyes, which is what peripapillary ischemia is. So the hypothesis for peripapillary ischemia is that we believe that the biofilms that are being secreted by the spirochetes, they are creating blockages within the blood vessels in the deeper parts of the retina. So we know the spirochetes are very small in size. They're only about a micron in size. So it's very easy for them to pass through small to medium sized blood vessels, and they can easily get into the central retinal artery. Now, once the patients are starting some treatment with, let's say, antibiotics, the white blood cell starts attacking these spirochetes, and spirochetes are building, or they are secreting biofilms and building larger and larger colonies. So once these colonies are small enough, they are traveling to different layers of the retina, but specifically deeper layers. And biofilms, once they are inside certain blood vessels, they can continue to grow and build into different larger size colonies as they are becoming more and more resistant to antibiotics. Now, the problem occurs when these biofilms are passed over to the plexus of the vessel surrounding the optic nerve head. Here, the biofilms can grow to a size of about 50 microns in size, which is extremely problematic because the blood vessel is only about 15 microns in size. And we believe this is what is causing these blockages of the vessels, leading to ischemia within the optic nerve head and leading to our finding of peripapillary ischemia. Another tool that we use in our office is the visual evoked potential. So visual evoked potential is giving us an overview of how the two visual processes are working. So this is an image of what a normal VEP should look like. So the N75 right here is the preconscious or the spatial system. The P100 is the response of the conscious focal system, and the N135 represents release from the response of the stimulus. This right here, the difference between the N75 and P100 is the amplitude change. So individuals that have that we are suspecting of having active Lyme infections or something going on underlying this N75 is going to continue deflecting all the way down into negatives. A normal finding is going to be typically around -4, -5 microvolts, but in individuals with active infections, we've noticed -15, -20. And then once they make this P100 response from the focal system, they are unable to release to get to this N135. So the P100 just stays up here. This is a very classic finding of what a overfocalization looks like. Another interesting finding we notice is that when the individual is just visually stuck up here, that also has some correlation to an individual who may have something like depersonalization or derealization types of syndromes. So there are a few different biomarkers that we've noted using the VEP. Just quickly here, this is what a normal VEP should look like. So this is an individual that does not have tick-borne infections. So this would be the N75, the P100, and then the N135 release happens, and then everything just plateaus out. This is one of my patients that has active Lyme disease. So initially they are very ungrounded. The N75 normal should be about -4 or -5. This patient is about -14 microvolts. They do create the P100, but again, this patient is just stuck up here. And this is what I meant by if they don't release this, they might have more and more neuropsychiatric symptoms like depersonalization. So this patient did in fact ended up having depersonalization because this patient was referred by their psychiatrist and they had similar signs and symptoms and findings of derealization. So what we do with these patients, again, we're using therapeutic prism lenses. So an individual that showed the N75 and then the P100 release did not happen. We added some therapeutic base in prisms, and this is the finding. This is how the visual process reacted to it. So the N75 significantly improved, and then the P100 also released. So this is as close to a healthy or normal visual process that we can expect to see within few minutes of adding a therapeutic prism treatment. So this was a extremely successful finding for this patient right here. So how to apply this, I'm just going to quickly go over a case report just to give you how to apply this information and what I can do as a neurooptometrist or what other optometrists can do. So here's another case report that I published just a couple of months ago. You can use the link here to access the article. So this I published in the International Journal of Infectious Diseases, the One Health Journal. This was a case about a 12-year-old patient who was referred by her neuropsych due to a lot of learning difficulties, dyslexia, developmental delays. By the time she came to me, she had already received plenty of therapy and rehab in general. Her main symptoms were blurry vision, headaches, a lot of difficulty with reading, visual processing dysfunction, avoidance of near tasks, difficulty tolerating busy, crowded environments, poor balance, coordination. She was starting to isolate more and more, major changes in personality and behaviors as well. These are the findings that are bolded here are binocular vision assessment in detail. And you can see a lot of them are showing out of norm findings, which is a very common finding that we see in most of our patients with tick-borne infections. Peripapillary ischemia again, so the ischemia around the optic nerve head in the left eye and ischemia around the nerve head on the right eye. OCT angiography, we're looking at the deeper layer, the retinal peripapillary capillary region in the right eye. For a 12-year-old, this should be all green in color. And we can see there's a lot of blue here in both the right and the left eye, showing that there is major vessel dropout here, which is, again, not a finding that I would anticipate in a 12-year-old. I could anticipate this in an individual who is, let's say, over the age of 60 or has some type of a retinal condition like glaucoma or some other retinal finding, but not in a 12-year-old. The VEP again showed a significantly deflected N75, and the release fully doesn't happen, so she's going up into these really high peaks here. Treatment with prisms showed a significant improvement just as soon as we added prisms. So this patient, again, we suggested running some comprehensive blood work, ended up giving her IGeneX testing kit, and she came back positive for multiple co-infections along with Lyme. I started treatment with prism lenses, binasal occlusion, treated her light sensitivity, and started a protocol for therapy as well. So just again, in summary, what I wanted to mention was, and hopefully you were able to get a quick summary of what we are able to do with many of our patients who have neurologic tick-borne infections. We can use a lot of our diagnostic tools and technologies using the N75 biomarker, peripapillary ischemia, but specifically not ignoring that there is a visual processing dysfunction that is leading to visual symptoms in these patients. So we have the tools and we have the capability of being part of the team for making sure these patients are being diagnosed early on and not developing complex neurological conditions. So hopefully you're able to, the takeaway would be the neurooptometric rehabilitation can be an important resource and tool for anyone working with patients with tick-borne infections. That's it.
Thank you, Dr. Sayyed. We are a little bit over time, but we do have a few questions. So if you're able to stick around, we'll
Absolutely.
start with.
I know I took longer than anticipated, so go for it.
Oh, we also have our poll question. Thank you, Nikki. So question number one in the Q&A, you mentioned cataracts as a condition of tick-borne infections. When treated, would there be any improvement, or is the damage for cataract progression, in this case, permanent?
So cataracts typically are not going to be the most common finding. Cataracts can occur due to, let's say, medications that the individual might be using, but cataracts is not going to be the most common finding. But with other findings that we are seeing as far as ischemia, we do see some improvements long term, but once the systemic condition is treated, yes, we do anticipate changes to occur, either slowing down the progression of cataracts or various other changes in peripapillary ischemia or visual symptoms in general. We do anticipate that, yes.
Thank you. The next question is, what was the test you said was used to confirm tick-borne diseases / Lyme infection? Does it distinguish between Borrelia burgdorferi, sensu lato / stricto?
So the two main things that we are using are visual evoked potentials in our testing and OCT angiography. So right now, all the research, all the work that we've done, there isn't an exact finding of, is it Borrelia, is it one of the other co-infections? So at this point, we don't have enough research. I think that's one of our goals, is to try to figure out what's causing which finding or which biomarker. But at this point, right now, we still have to work with the physicians or our other doctors that we are co-managing with to find exactly what is active. Our testing can give us red flags or give us some inclination that there might be something active, but we don't exactly know what the infection would be.
And looks like our last question here, did you work only with Lyme disease or other tick-borne diseases?
So it really depends what the patient presents with. So we do see Lyme disease, but we see all other co-infections as well. Just this morning, I've already had two different patients from outside of Connecticut that came in with multiple co-infections. So a lot of them are going to present similarly, but we do obviously notice that sometimes patients have, let's say, more light sensitivity, or they may have what's called visual snow or visual static, which I do think there is some correlation with specific co-infections. But honestly, I think we still need a lot of work and a lot of studies to figure out how to make those correlations so we can further focus on the more specific treatment plan.
Thank you. But this will be the last question. Sorry. How long do these symptoms last in patients without active Lyme infections?
So it depends on the scenario. So anyone that's coming in with neurological symptoms, either it's tick-borne infections or concussions, TBIs, the brain process has to be treated. So if it's not treated, it's possible that the patients can continue suffering with visual symptoms the rest of their life, because there's a lot of accommodations that they're making while they're sick. So unless the brain process is changed or we give them some support in bringing balance back to their visual process, they could potentially live with those symptoms, and that could be their norm. But that's why so many patients come to us even outside of state, because they've tried everything and nothing has worked, because everything has been a little bit more superficial rather than going after the brain process rehabilitation.
Great. Well, thank you for presenting, and thank you all of you for your questions. I do just real quick want to talk about tomorrow a little bit, and then I will send you on your way. We started from the beginning. Okay. Can everyone see? This is tomorrow. We're back to our schedule here. So again, if you didn't know, we have more of the Tick Academy tomorrow. It will start at 10 a.m. Central Time again. And we have all of these great speakers here. If you're not registered for some reason and you're available to attend, please scan the QR code or register at the link there, which is bit.ly/TICAcademy24. And tell your friends. You know, the more the merrier. I want to again thank all of our sponsors. Without their support, this would not be possible. And we really appreciate their help. And again, our primary funding is from the North Central IPM Center. And we are currently in the process of applying for next year's grant cycle. So if you liked what you saw here today and want to help support the work of the TIC IPM working group, please consider writing us a letter of support. If you would like more information on this, please send me an email. And I would be happy to give you our template or just tell you more about the working group. So thank you all so very much for attending today. I hope that I will see you tomorrow. And tell a friend. So thank you very much.